It seems. Doug, please, we can't see your screen. Oh. I think it's off.
Hello. Please, can you see the screen?
Doug, you can. Doug is off again.
Yeah, I think a lot of your colleagues are not in class. Mm. People went for hangover, sleepover, and they've not returned yet. Yeah.
Yes, chilling over too.
So we'll wait for a couple of minutes and then wait for a couple of minutes. Then yeah, a lot of folks went for hangover, sleepover, and they just don't want to wake up. And chilling over, and especially the men. And because my ladies are all in class, where are the men?
Doug, this one, your lady, so
Oh,
the
my
men here.
ladies are in class.
The
I
men
can
are
see
here. Most of the men are here.
We did.
oh,
Don't
my ladies.
come to class.
My ladies are in class.
Yeah, let's count the men to the ladies
The
ratio.
men,
Roll call.
the men,
Yeah,
Doc, roll call. Roll call.
the men went for. I'm waiting for my ladies to, and then I will before I give the roll call because I don't want to
No,
say shall
look at them. Look at
give
them.
up. It's not fair to my ladies.
Hey, X-Man bias. X-Man bias.
Oh, see
X-Man
you. You know, in this world, everybody and his or her own sickness. Me, I'm suffering from men blindness. You know, people suffer from night blindness. Others have river blindness. Others are short-sighted, long-sighted. Mine too is men blindness. When men are there, I can't see them. I can't hear
Can't
what
see them. Yes, you can only see the ladies.
I can't hear them. I can't see them. Oh, Charlie, it's not me.
Doug, you can hear them.
have to have to ask God so that God will heal me. Before we enter into the next year. Please put it on your platform. Put it on your platform. I'm sure some of your colleagues, once they get to know that, Eben just joined. Yes. The men are coming home because we are not even up to half of the class. Our year class, we are not even up to half. Okay, so please let them know that we are here and we want to start. I don't want to miss anyone. Okay.
Hello, sir. Sir, please, are you sharing your slide?
Yeah, it's my slide. I think last time I gave the slides out.
Oh, I thought you are sharing the slides now.
Yeah, the slide is already shared. The screen is shared. It's not appearing in
I'm not.
yours.
No, we
I'm not
can't see
seeing
it on the
anything.
floor.
It's on the screen, or maybe your network.
Oh, we can't see. We can't see.
No, we
Okay, we
can't see
can't see.
anything.
It's not
You can't
showing
see the
any.
screen.
Yeah,
Yes, we can't see anything on it.
yeah, it's not. It's
All
not.
right, sir.
We
You
can see
cannot
it
see
too.
it. You can see it
I can
now.
see it too.
No,
Yeah,
no,
it
it's
has
back.
not come. It has not come.
It just came. It just came.
I've
The
been
men
seeing
can't see. The men can't see. Some of them can see. You see, they are now people are now washing their faces. So you see,
As
from the hand
usual. As
over,
usual. As
you see
usual.
your life. You see your life.
Hello.
You can go and do whatever you want to do. Now, the following day, the screen will betray you. Eh? The screen will betray you. Eh? The men. So
Their
the
their
hand,
devices are their
the hand
devices are just
opening is
warming
over.
up. The devices are warming up.
Oh, you see, oh. People just added the water and they charged the one the yesterday one, and the screen disappeared. Eh? Eh, okay, let's see.
This
Let me
one.
give let me give shout out to my ladies, okay, because they've been waiting for the men for so long. Okay, so let's see. Little bit of shout out. Johnny, okay, I can see my Johnny. Aaron Kofi, yes, welcome to class. Afishapa, Alexi, okay. Oh, my one angel, Angela, good to see you. Another angel, I have two angels here, okay. Angela, two Angelas, okay. I see you all. It's nice seeing you in class. Kwame Bright, good to see you. Charity, you are there. I see you. Vincent, give a wave of to the Lord. Eben, I see you as well. Kwekumensa, I see you. Lizzie, one Lizzie. Kumsin, I see you. Esther, oh, give a wave unto the Lord. Okay, God has been so faithful from the beginning of the year. Oh, that's nice. Esther, I see you. Emil, I see you. Gilbert, yes, you are there. JJ Lawrence, okay. JJ Lawrence, okay. Your name is close to JJ Thompson, okay. All right, good. And then Johnny, I see you. John Sewa, I see you. Nana Che, I see you. Kwame Afriye, I see you. Nicholas Baden, I see you. Peter, I see you. Peter Ade, I see you. Ritual, I see you. Oh, one rater. Give a wave offering unto the Lord. Give a wave offering unto the Lord. Samson, I see you. Sammy Abaka, I see you. Okay, Silas, I see you. Oh, Tampori, I see you. Good to see you. Piero, Peter, I see you. Really, I see you. Abdallah, I see you. I see you. Zachariah, I see you as well. I see you all. Welcome to class. Oh, my one Nimatu is back in full swing. My one Nimatu is back. Okay, unstoppable. Okay, she's back in action. All right, she just woke up, but that's okay. She just woke up. That's okay. All right, okay. Folks, we have ourselves. Nana, why are you laughing? Why are you laughing? Please don't do that. Don't do that to my one name too. Okay. Good to see Esther, your boy. Welcome. Okay, I see you. I see you. I see you all. Okay, that's good. Good to see you all in class. Okay. Good to see you all in class. Good to see you all in class. Okay. Good to see you all in class. Good to see you all in class. Good to see you. Good to see you. Mary, I see you. Okay, Mary. Mary, Jesus' mom. This season is your season. Okay. Thank you for giving birth. Okay, to Jesus. And then people are now, the men are passing over Jesus to do hangover and sleepover. Eh? Somebody born in sand. The men are taking advantage for hangover and sleepover. They're still not waking up. Eh? The men, they will make Jesus bowl. Eh? Jesus and Mary go bowl. Okay, so I think in a few minutes' time we are going to. Okay, can't be waiting forever. Please share the link with your colleagues. Let them know that Kwame is here. I'm waiting for them. Okay. All right. So we've not gotten half of the class. Oh, because we are expecting close to. Okay. Well, maybe folks will join pretty soon. So okay. So the last time we started with cell, and then we talked about the history. In fact, the cell biology is who gave birth to cell biology, and then after we talked about who gave birth to cell biology, which is microscopy, we delve into the world of microscopy a little bit, and then we talked about two main types of microscope. Okay, let's see. Let me take blank page and see, we try and see we can do recap from a blank page. Okay, so we said that the last time we said that the cell biology, microscopy, okay, give birth, okay, birth to cell biology, and we went on about the history of microscopy, okay, talked about the history of microscopy. Then we talk about compound, two types. We talk about the optical. Yeah, the optical we talk about. We have the simple and what and compound microscope. Then we talked about electron microscope. Then we talked about the three terms associated. Three terms. The three terms used to describe microscope. That is the contrast. That is the magnification. Head resolution. Okay. Resolution. Then we also talked about size of cells, and then based on that, we talk about surface area to volume ratio. Okay, and then we talked about levels of what? Levels of organization. Okay, so this is basically a recap of what we did the last time. Okay, so today we'll take those stuff one by one and then we'll talk about them. Okay, we need to, honestly, we need to finish up today. We have 4 meetings, but we need to try as much as possible. Cell is quite plenty. So we need to, and this part of the subject is very, very important because at SS level, that's one of the things that the teachers, especially the genetic aspect, so the teachers tend to struggle to deliver those ones. So we need to master them. Okay. All right. So I think our colleagues will still join. It's top of the hour, so I know folks will join as we move along. Okay. All right. Good. So today we're going to talk about plant cell and then the animal cells, and then we'll talk about organelles and stuff like that. Okay. Good. So I don't want to waste a lot of time here. So let's start with okay. Let me start with okay. So let's say we have something like a word. So based on maybe cell complexity, okay, based on cell complexity, we can have two main types of cells. Okay. Let's see. Let me go to design and then split my slide. Let's see layout. Design. Oh, slide. Design. Let's see layout. Okay. All right. So let me split my slides into two to make life easy. Okay, maybe something like this. No, this one doesn't allow me. Okay, so let me take this one. Let me take something like this. Okay. All right, great. So we'll talk about cells. Okay, and then okay, great. Let me put this one here. Now, in terms of cell complex, if you ask me to classify cells, okay, based on what? So cells, so can get types of what? Types of types of cell, okay, based on what? Based on complexity, okay, complexity. So if you base it on complexity, we can get to what? We can get prokaryotic cell, prokaryotic cell. I think we talked about it the last time. And then we'll talk about, so this prokaryotic cell, this one refers to what? Bacteria, bacteria cell, okay, prokaryotic. And as I said the last time, the pro means before, karyo means nucleus. So it's more or less before nucleus, okay? And then we can also talk about what? Eukaryotic cell. In eukaryotic cell, we can talk about the plant cell. Okay, we can talk about the fungi cell, the fungal cell, and we can talk about what? Animal cell. Okay, like that. So these are eukaryotic. And eukaryotic, the word "eu" means even or true. Okay, in Latin. So eukaryo means true nucleus. Okay, right. So these are simply true nucleus. Okay, nucleus. Right, and then this one is a before nucleus. Okay, all right, good. So these are two main types of cell. Okay, so we'll take them one by one. And let's start with prokaryotic. Prokaryotic cell. Prokaryotic cell. Prokaryotic cell. Okay, now when we talk of prokaryotic cell, please, bacteria cell. What are the structures that actually come into mind? When you say a cell is okay, we know it is bacteria cell, but what are the things that actually come into mind? Yes, can somebody help me? Prokaryotic cell. Yes, let me go to my audience. Okay, prokaryotic cell. Yes,
So please, this
please.
is fine.
Yeah, yes, please. Let me hear from you, Johnny. Johnny, please, can I hear from you, please? When you say prokaryotic cell, what is it?
So this is Sammy.
Sammy, oh, Sammy, okay, Sammy, let me hear from you, please.
So please, can you talk of the mitochondria?
Okay, prokaryotic cells, they lack mitochondria. Okay,
Okay. Okay.
yes. So, okay, let me see if I don't have the picture here. I have to let me see whether I can get a beautiful picture for you. I can. Do you know what? I can make a proper sketch. This is. I can. Sometimes life is better when I do it like that. Okay, so let's see. Let's see whether I can make a rough sketch like this. So let's see something like this. Okay, like this. Because as I sketch, you can also sketch as well. So this is okay, and then we have something. Another let me draw another layer like this, another layer like that, another layer like that. Okay, good. And then so let me call this last layer here. Let me call this last layer here cell membrane. Okay, so let me call this one cell membrane. Okay, and then there is this structure here, this outer part here, we call it what? Mucilage. Okay, so you call it mucilaginous. Okay, mucilage. Okay, mucilaginous capsule. Okay, I'm using my marker to write, so it's a little bit slow. Okay, mucilage. So it's mucilaginous capsule. And then beneath this mucilage, there's another layer here. There is a cell wall. Okay, there is a cell wall. Okay, this cell wall, but this cell wall consists of something we call peptido, pepti, peptidoglycan, peptidoglycan. Okay, great. So we have the mucilaginous capsule. Beneath the mucilaginous capsule, we have a cell wall. Okay, all right. And this cell wall is made up of what? Of a structure. We call it what? We call it peptidoglycan. So pep talks about amino acid, combination of amino acid protein, and then the glycan talks about carbohydrate. Okay, so we have this special cell. We see in plants, the cell wall there. When we talk of the cell wall in plant, it's made up of cellulose. But when it comes to the cell wall of what? Of bacteria, it's made up of a structure we call peptidoglycan. Okay, all right, good. As a matter of fact, okay, this is not really diversity class, but let me just throw a little bit light on this one. You know, this peptidoglycan is very, very important. In fact, bacteria cells can be grouped into two based on the thickness of the peptidoglycan. Okay, so we have the gram positive bacteria and we have gram negative bacteria. Okay, so okay, you let me finish labeling the parts, then we'll come to it. Okay, then let's say inside here, they don't really have nucleus. So you see, they are chromatin material, those genetic material. Okay, this one, they are chromosome. Okay, they are scattered. So here, what I have here is the word, is the DNA. Okay, that's a DNA, or maybe I can call it chromatin. Okay, chromatin material. Okay, chromatin. Please, as I draw, please, you can draw chromatin. Chromatin. Okay, this is the chromatin material. Okay, great. So chromatin. And then we have this thing, we have cytoplasm here. So so let me see cytoplasm here and cytoplasm. And then there's one particular thing that they must have. There's this organelle they must have because so this small organelle. Okay, so let me point one of them and I'll call it ribosome. So ribosome, they will have ribosome. We have ribosome. Say ribosome. Okay, and then maybe the last thing that I'm gonna talk about. So they have ribosome, okay, and then so they have cytoplasm, and then there is this genetic material, DNA chromatin. A cell wall consists of peptidoglycan. They have cell membrane, and then um what is the last thing I like to highlight? So they have ribosome for protein synthesis, for protein synthesis. Protein synthesis, okay, to synthesize, okay, for protein synthesis, okay. And maybe the last structure I will add it here is that at the base here, they have this long hair-like structure, flagellum coming out of it. So they have a flagellum, 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 okay, for where? For locomotion, for locomotion, if that's movement. Okay, good. So this is more or less a generic picture, okay, of DNA of prokaryotic cell. And one thing you should know is that okay, the this DNA that they have here is not linear like ours. You know, we have linear DNA. Even though I've drawn it round like that, they have so this is what is forming the chromosome. Okay, so you know the DNA will come together to form the chromosome, which I'm referring to as chromatin. But in the case of bacteria, okay, this DNA thing is secular. It's not linear. Okay, it has secular. Okay, so this DNA is what secular, and it's something unique. Okay, it's secular. Okay, the DNA here is secular. Okay, you know the DNA that is forming the chromosome is not what? It's not linear. It is what? Secular. Okay, and then occasionally they may also have this round structure. Okay? So what is here is called genomic DNA. Is genome. Okay. So what is here is called meet your mic. So here this is genomic DNA, genomic, okay, more like okay, genomic DNA. And occasionally some of them tend to have extra DNA, extra this small round DNA here, which we call plasmid. Okay, plasmid. In fact, plasmid, what they are actually involved in the survival of bacteria. Plasmid. Okay, plasmid. So in fact, for bacteria to become resistant to antibiotic is usually due to the presence of the plasmid. Okay, so this is general overview of that. You know, there are some structures. You see, if you look at my diagram, even though it's not so beautiful, there are some organelles that I've written here. I've written ribosome. I've written I've indicated the cell wall, which is made up of peptidoglycan. I've talked about the circular DNA. I've talked about the flagellum. I've talked about the mucilaginous capsule. Okay, I've talked about the cytoplasm here. Okay, but there are some things I didn't mention. Look at that. I already made mention of what. Okay, now you see, look at this structure here. I didn't mention mitochondria. Mitochondria, you don't have it. Mitochondria. There. Okay, they don't have so they lack what? You see, so look at the things I'm going to, so they don't have chloroplast as well. Okay, chloroplast as well. Okay, they may have chlorophyll. Okay, there is this cyanobacteria. Okay, there is this bacteria, we call it this prokaryotic cell, this one, bacteria or cyano, let me add it here, cyanobacteria. Okay, cyanobacteria. So now cyanobacteria contains chlorophyll, okay, but it doesn't have chlorophyll. It has a pigment. The pigment that absorbs light for photosynthesis, it has it, but it doesn't have chloroplast. Okay, so why look at the what I'm showing? It doesn't have Golgi body, Golgi, Golgi body. It doesn't have lysosome. Okay, right? It doesn't have endoplasmic reticulum. Okay, now, so all these things, so these are organelles that prokaryotic. It doesn't have nucleus. That's very important. Nucleus. So organelles that prokaryotic cells lack. Okay, good. Now, what can you tell me about these organelles? There's something about them. These organelles that I've mentioned, that all of them, we are saying that they are absent in prokaryotic cells. What can you tell me about them? Look at the Is anybody from the audience? What can you tell me about them? Yes. What can you tell me about them? Yes. Yes, please. Go
So,
ahead. Yes, please.
so
Um,
I can do something about on the chloroplast.
yes, please.
The chloroplast is the organ that produces chlorophyll.
Okay. Okay. I'm not even talking about you talking about it individually. I'm talking about
Okay.
collectively. What is
It
common?
is okay.
Prokaryotic
Then,
cells or bacteria cells, they lack all these things that I've written. And
okay.
what is unique about all of them? I want one sentence that will talk about that will describe all these ones.
Okay.
Okay.
Then I'll come again.
Okay, no problem. Yes. Anybody?
Please, we are membrane-bound organelles.
My ladies, where are you? Yes. What is it unique about all these organelles that I've written there? Yes. Good. Oh, Helena, good to see you back. Helena has nailed it beautifully. Okay, beautifully. Well done, Helena. Oh my God, my ladies are back. Somebody should call fire service, please. My ladies are on fire, please. They should call fire service for me, please. Okay. So I can even add this one. That's a peroxisome, all these ones. Okay. One thing about all these organelles that I've written, you can see that they are all membrane bound. Okay, these are membrane bound organelles. They are organelles that are surrounded by membranes. So one unique thing about prokaryotic cell is that okay, they lack what? They lack they lack they lack membrane bound organelles. Are you with me? Okay, some will say that they lack nucleus and membrane bound organelles. Okay, but nucleus is also one of the membrane bound organelles. So I'll put them together. Okay, good. So they lack one unique thing about prokaryotic cells. Okay, is that they lack what? Membrane bound organelles. The organelles are not bounded by membrane. So all the membrane bound organelles like this one, all these ones that you've written here, okay, they don't have it at all. They lack all of them. And these are all membranous organelles, the organelles that are bounded by membranes. And prokaryotic cells, they lack them. Okay, all right, great. Anything more?
Doc, please.
Yes, please.
At the initial stage, you made mention of, or you talked, or you asked a question about some of the organelles that are
Oh,
located on
I was
your
not
protein.
interested. I was not interested in individual organelles. I was
Okay,
interested in. I was interested in what is common to all these ones. That's my what I was. Okay.
okay. So
Yes. So
no,
yes,
please, with the
yes, please.
prokaryotes, we know the powerhouse of the cell is the mitochondria,
Yes,
and that
please.
enables the cell to perform whatever activity that you want.
Cellular respiration. Good.
Yes,
Good.
yes, yes, and stuff. Now that the prokaryotic cell lacks this mitochondria, what is the powerhouse? How does it carry out its
Beautiful.
activity?
Beautiful. I know your wife asked you to ask this question. Such a brilliant question. Well done. Nice question. Good. Okay, now you see, this mitochondria is the powerhouse. What does it do? Why are you calling it the powerhouse? It generates energy in the form of ATP, right? Okay, so most bacteria, because you see, when it comes to cellular respiration, which we will talk about it, but because you've asked this question, let me just take the opportunity to explain here. Now, this is the powerhouse of a cell. Okay, we know that. And it produces energy in the form of what? ATP, adenosine triphosphate. Okay, and this ATP, now, so the question is this. Now that we have this ATP here, okay, we don't have mitochondria. How is the ATP generated? Now, when it comes to cellular respiration, okay, if you come to cellular respiration, there are three main processes that we talk about, okay? So when you talk of cellular, cellular, okay, cellular respiration, okay, there are three things we talk about, okay? We talk about one is glycolysis. Glycolysis, okay, glycolysis doesn't require oxygen. Okay, followed by something we call Krebs cycle. All these we shall come to it maybe along the line. Okay, followed by a Krebs cycle. And then the last one, if you call that one electron transport chain. All right, great. Now, glycolysis is simply splitting of lysis means break. So glyco talks about glucose. So glycolysis is simply breaking down of glucose to release what? To release to the net ATP. That will be net ATP. In fact, glycolysis, when glycolysis is occurring, it spends 2 ATP, but at the end you get 4. So which means that the net ATP produced from here, net ATP is just 2. So 2 net ATP you produce. All right, good. Now these ones here, okay, they actually occur in where? In the mitochondria. This one will occur in cytoplasm. Glycolysis here will occur in cytoplasm. It doesn't depend on oxygen. So it can occur whether oxygen is present or oxygen is not present. Okay, it can still occur. And then we have the Krebs cycle and electron transport chain that will occur in the mitochondria. Okay, all right, good. Now listen to me here. So in most bacteria, okay, or prokaryotic cells, they rely on what? They undergo just this one. They undergo this one, which will occur in the cytoplasm. That does not require Krebs cycle and electron transport chain. So they will just go with this one, just this one, glycolysis. Okay, so they undergo, so this glycolysis doesn't require oxygen. So we call it what? If it doesn't require, we call it anaerobic respiration. So they will undergo anaerobic, anaerobic word, respiration, which is the glycolysis. Okay, and remember it's okay word. Okay, it will be in the word cytoplasm, and then doesn't doesn't what require oxygen, doesn't require O2, and then produces net ATP of 2. A net ATP of 2. Good. All right. So these are anaerobic bacteria. So most bacteria tend to rely on anaerobic respiration, which is just simply glycolysis. Those, some of them can undergo aerobic respiration. Ooh, that's a beautiful question. So those that can undergo aerobic respiration, what happens is that this Krebs cycle here, all right, will occur in their membrane here, the cell membrane. Okay, so in their cell membrane, okay, there is this okay, in their cell membrane, there is this structure. So let's say those ones, okay, let me take a marker, let me even take a blank page and draw. Okay, in their cell membrane, you see the cell membrane goes like this. Okay, the cell membrane, their cell membrane goes this way. If this is the cell membrane, you see the inner part of the cell membrane goes like this, it's folded. Okay, remember that in the mitochondria, the inner part is folded to form something we call cristae, which we shall come to. So this inner part of the cell membrane of the bacteria that exhibits aerobic respiration, the inner membrane is folded. And this inner membrane, okay, this folded inner membrane is what will be used for aerobic respiration. So those bacteria that tend to undergo aerobic respiration, that is a respiration that requires oxygen, respiration that requires oxygen, they will use they use their cell membrane, okay, membrane for the Krebs cycle, and electron transport chain. Please, are you clear? Hello?
Yes, sir. Sir, I'm here.
Good. Do you understand it now?
Yes, sir.
Thank you.
Doctor, I'm okay.
That's a nice question. That's a very brilliant question. Okay. I know your wife told you, but I've forgiven you. Okay, good. That's a very brilliant one. Okay, I know. Good. That's a very nice question. Well done. So I've even taken the opportunity to explain this one. So, but prokaryotic cells, they are simple compared to, so they are the most simple cells. So they are more or less the simplest of cells. Okay, they are the simplest of what? Simplest of cells. Okay, they lack membrane-bound organelles. Okay, and then we are referring to the bacteria. Okay, and the cyanobacteria. And then most of them, because they lack membrane-bound organelle, our brother asked, then if mitochondria is the powerhouse of a cell, what is it? So the cellular respiration, they are in three phases: glycolysis, Krebs cycle, electron transport chain. But because, and this one doesn't require oxygen to okay, so almost all organisms seem to exhibit it. The ATP produced not all that much too, but this is a very small organism. So net ATP of 2 would be cool for this particular organism. Okay, but those that tend to demand oxygen, they use the cell membrane. The cell membrane is the inner part of the cell membrane is folded so that Krebs cycle, and then so that it will have the enzymes for Krebs cycle and electron transport chain to work. In fact, electron transport chain is a part that is where most of the ATPs are produced. That is where most of the ATPs are produced. Okay, beautiful. So we know that prokaryotic cell, which we are referring to, this my scattered diagram here, the mucilage, genus capsule, the flagellum, the cell wall consisting of peptidoglycan. The cell wall is not made up of cellulose. It's also peptidoglycan. It has circular DNA. The DNA is circular. Okay, circular DNA, very important. It has something we call plasmid. And this plasma, as I said, it helps the survival of the bacteria, okay, especially when it comes to insecticide resistance and then survival in harsh condition. It is the work of the plasmid. And then we have the cytoplasm here. And then there's this particular organelle that no matter what, it will possess. Okay, so that is a ribosome, which is used for what? Protein synthesis. Okay, great. Beautiful. Now, do you know what? We believe that scientists, okay, we believe that, okay, our modern day Okay, so let me maybe there's a picture here, even though I've drawn it. Okay, so there's a picture here that you need to see. Please, this one is you see that they say that nucleoid region. Okay, so the space where the nucleus is occupying is sometimes referred to as nucleoid, but that's not nucleus. Okay, it's just because the DNA is there, that is why it is called nucleoid, but that's not nucleus. All right, good. Okay, and then some of them have this sort of the pili, the hair-like structures originating from the wall, pili. Okay, all right, good. So this is the generic one that I drew for you previously. Okay, okay, the generic one that I drew for you. All right, okay. So we have this one here. Okay, so beautiful. Okay, great. Now, the what we believe is that there is this scientist. Okay, let me show you the picture here. Okay, Lynn Margulis. Okay, Margulis actually came with a certain theory. In fact, she is not really the origin of the theory. Okay, there was this Russian guy, his name is Konstantin. Okay, all right, I do. It's not history class, I won't bother your head about it. Okay, Konstantin actually was the first person to come with a theory. Okay, all right, good. And then what is this theory about? Okay, so there's something called endosymbiotic theory. Okay, but before that, let's go to eukaryotic cell first, and then before, okay, let me talk about eukaryotic cells first. Okay, eukaryotic cells. So eukaryotic cell, these are cells, they have what? They have membrane bound organelles. Okay, so they have definite nucleus with what? Definite nucleus, and then also have what? Have a membrane bound organelles. A definite nucleus with membrane bound what? Organelles. Okay, great. So all those stuff that we talked about that they are lacking in what? So which means that they will have the mitochondria. They will have the let me just copy them. Okay. They will have they will have all these ones here. Okay. Okay, they will have that. So all these organelles are there. Okay, great. Okay. All right. So here, when we talk of the eukaryotic, as I say, we are talking about the plant cell, fungal cell, and what? Animal cell. So these are the eukaryotic cells. Okay, these are eukaryotic cells. Okay, eukaryotic cells. All right, great. So with these eukaryotic cells, what can we put here? Now, if you look at the size of these two chloroplasts, okay, if you look at chloroplasts, okay, all right, let me bring a picture of it and then show you something. Let me get a picture of it. Let's say chloroplasts, okay, so that you get fair idea of what I'm talking about. Okay. Okay, let's see. Good. So let me get you. Okay, so let me take the chloroplast here and then bring it here. Okay. Then let me take the mitochondria here. Okay, so let's see. All right, so I have my chloroplast here. Okay, so chloroplast, you stay here and wait. Okay, let me bring the mitochondria. Okay, let me take mitochondria as well. Okay. Let me take mitochondria as well. Okay, so let's say on a simple diagram of mitochondria. Okay, okay, great. So let me get this image as well. Okay. All right, mitochondria is here. Okay, so mitochondria, you come down here. Okay, so you go up a little bit. Okay, so let me shrink the size more. And then bring this one here. Okay, then you know something? Let me bring the bacteria. Let me just get some bacteria. So then probiotic cell structure. Okay, let me take the one that a little bit close. Okay, good. Okay, so let me take this one in a copy. Okay, well, let me take this. Okay, all right. So let me copy that and then bring it here. Okay, so let's see something here. All right, great. Okay, so let's see. Now let me put this thing side by side. All right, I think it's better I take a new slide rather and put them there. Okay, that will make life easy for all of us. Okay, let me cut this one. Okay, so you come here. Okay, so come here. Let's see. We bring this one here. Okay, you come here, and then let me bring the the chloroplast here. Okay, mitochondria to here. Okay, all right, good. So great. Okay, all right. So great. Okay, all right. Now, so if you will look at this one, you can see that this bacteria here and this they have, let's see the things they have. So they have well, let's see, okay, the things that I want them to label. Okay, you can see there's ribosome here. Do you know what? Let me get chloroplast with better label. I know. Let me get chloroplast with better label. I'm doing that deliberately. Chloroplast with better label. Okay. All right, good. So let's see. Okay. All right. So let me take this one rather. Okay. Let me take this one. What is here is correct, but I want the one with better label because I want you to see something. Okay. So let's see. Okay, great. Now, all right, great. So let's see this one. All right. Okay. Uh-huh. Now, let's put these three things there side by side. Let's see something. This one has what? It has circular DNA. Okay, circular DNA here. All right. This one also has what? Has circular DNA. Circular DNA. This DNA here is also circular. Okay, in the mitochondria. This one has ribosome, right? Where is the ribosome here? There's ribosome in there. So this one is the mitochondria. There's ribosome there. And there's also, there's ribosome here. Okay. So what scientists believe is that, and if you look at the size of the bacteria cell, the prokaryotic cell, okay, the size is almost comparable to what? To chloroplast and what? Mitochondria. Okay. All right. It's almost comparable to what? Chloroplast. Oh, I've taken this one again. I didn't take the. I've taken. Oh no. Come in. Chloroplast. Chloroplast. Ribosome. Okay. I think. Okay. There's this simple annotated diagram here. Let me pick this one. Okay, so okay, that's great. Okay, so this one is simple diagram. Good. Now, if you look at these three structures, you can see that all of them tend to have circular DNA. They all have ribosomes. They all have so they have ribosomes. And then if you look at their size are what? Are comparable. You have comparable size, okay? You have comparable size. And with this comparable size, okay, what happens is that, okay, so they have the comparable size, okay, great. All right, so they have comparable size, all right. And with this comparable size here, what is going to happen is this, okay? What is going to happen is this. If you look circular DNA, circular DNA, circular DNA, mitochondria, 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 and then the size are comparable. So what the Russian scientist, his name is Konstantin, but we don't need to go that fast. Okay, good. Okay, they brought something theory we call endosymbiotic theory. The reason we talk about Lynn Margulis is that Lynn Margulis was the one who provided empirical evidence in the lab to say that, which means that our modern day eukaryotic organisms. Okay, so what he's saying that he brought a theory called the theory of what? Symbiotic theory. Okay. Now, when you say symbiosis talks about biological association, okay, existing among organisms. So, like mutualism. When you say symbiosis, we are talking about mutualism, commensalism, amensalism, those things, parasitism. Those are all symbiotic relationship. Now, so we have something called endosymbiotic theory. So then, according to this endosymbiotic theory, what is stating is that our modern day, okay, so our modern day eukaryotic organism actually came into being from symbiotic relationship between prokaryotic cells. So what it means here is that in the olden days, there were these prokaryotic cells like this. Let's say mitochondria is here, okay, chloroplast is here. Chloroplast and mitochondria, they used to live as free bacteria. They were free living bacteria cells. And eventually, two of them are all associated with energy. Mitochondria breaks down glucose to release energy in the form of ATP. And then chloroplast uses sunlight to build up glucose. So all of them are associated with energy. So what happened is that, Lynn Margulis in 1970 provided evidence, okay, the empirical evidence in the lab to support this claim that our modern day eukaryotic organism that we see with the nucleus, with the mitochondria and everything, okay, they form from the word, from the amalgamation of what? Of prokaryotic cells, meaning that different prokaryotic cells came together to form the what? To form the eukaryotic cell. So today we have a eukaryotic cell like this, okay, with the mitochondria, with the chloroplast, with the nucleus, with the Golgi body, with the ER. Okay, so according to this theory, what this theory is saying here is that this modern day eukaryotic organism, they came into being through amalgamation of prokaryotic cells. Meaning that prokaryotic, so this mitochondria used to be a free living bacteria, okay, and that chloroplast was also a free living prokaryotic cell, and eventually all of them came together through symbiotic. Please mute your mic. Mute your mic. Please mute your mic.
Madam Mercy, mute.
Mercy, mute your mic, please. Okay. So they came together as a result of what? Amalgamation. So it's just like in your family. Okay, let's say right now, your family now you have in, okay, is the word, is the your nuclear family that you have in is representing eukaryotic cell. Okay. Now, what we believe is that the wife used to be a free living prokaryote, the husband free living prokaryote, and maybe let's say the kids also free living prokaryote. Okay, and you all came together. Okay, you entered into a symbiotic relationship whereby one organism captured another organism. Okay, you captured another organism, and when that organism was captured into this one, then it resulted in what? It resulted in the formation of what? Of the eukaryotic cell. So our modern day prokaryotic cells, okay, so what this theory is saying is that, okay, what this theory is saying is that, okay, the modern day eukaryotic cells, eukaryote, okay, cells come from where? They come from amalgamation, okay, amalgamation, okay, or come from a symbiotic, okay, symbiotic association of prokaryotic, prokaryotic cells, okay, come from amalgamation of prokaryotic cells. Right, good. So with this one, with this amalgamation of prokaryotic cells, so what it means here is that the way we can look at this thing is that if you look at the membrane, if you look at this one, the membrane here, okay, the membrane surrounding the chloroplast and the membrane surrounding the mitochondria, they are similar to the membrane surrounding prokaryotic cells. So the prokaryotics, if you look at the membrane surrounding them, and the membrane surrounding mitochondria, and then the chloroplast, they are similar. They also have similar ribosomes. Okay, so they have similar ribosomes. So what are some of the evidence? Okay, evidence to prove that. So the evidence supporting is the fact that, okay, they have words. So similar ribosomes, okay, and then they also have similar ribosomes, and they also have what? Circular DNA. Okay, so the one who provided evidence to support the endosymbiotic theory in 1970 is Len Margulis. Okay, it's a brilliant, it's a American evolution biologist, evolutionary cell biologist. Okay, very brilliant, very smart lady, very smart person, very brilliant. Okay, so she came out with the other reason, but I don't at your level. This is basic cytology, so I don't want to go into too much details. Okay, but the other reason, but the ones you shouldn't forget is the similar ribosomes and then what? Circular DNA. So which means that the ribosomes that you find in mitochondria and chloroplast, they are similar to that of what? They are similar. They are what? Are similar to that of what? The modern day prokaryotic cells, right? So similar ribosomes and similar circular DNA, they are all confirming that our modern day eukaryotic cells, they came as a result of what? Symbiotic association between among prokaryotic cells. So the endosymbiotic theory is just simply saying that, okay, the endosymbiotic theory is simply, so endosymbiotic theory is simply, endosymbiotic theory is simply saying that, so in this biotic theory, okay, it's simply saying that eukaryotic cells, okay, okay, arose from exchange with exchange eukaryotic cells. And the evidence is what? Mitochondria and chloroplasts. Okay, chloroplasts, mitochondria and what? And chloroplasts. That seem to have similar structure to our modern day eukaryotes. Okay, and then the one who brought that evidence, is American scientist called what? Lynn Margulis. Okay, in 1970. Any headache so far? Any headache so far? Is anyone's head spinning? Okay, so now that we know all this one, okay, we're gonna take the other parts one by one. I think I've explained this one already. I don't have to worry your head about this one. Okay, good. So we're gonna talk about plant cell and what? Animal cell. Okay, and after that we'll take the organelles one by one. Okay, and talk about them. Okay, good. So let's see. Let's talk about our this eukaryotic cell. When we say eukaryotic cell, we know that they are cells that are bounded by what? They have membrane bound, they have definite nucleus and membrane bound organelles. Okay, all right, great. And then so in fact, all eukaryotic cells, you have nucleus, you have membrane bound, and you have what? Cytoplasm with organelles. So we're going to take them one by one and then we'll talk about them. So as you are, let me take blank page again. Okay, let me look for blank page. Blank page. Yeah, okay. Let me pick this one. Okay, so like this, so something like that. So let's say we have so let's start with plant cell. Plant cell. All right, great. Okay, so plant cell. Okay, please, I want you to draw. So occasionally I do ask it in exam. So I'll let's see whether I can get nice plant cell structure for you, and then I'll use it to explain what I want to talk about. Okay, so let me pick this one. Okay, let me see what let me pick this one here. Simple plant cell structure. Okay. All right, let me copy this one for you. All right. Okay, so let's see. So please, I expect you to draw. So, all right, good. So let me put this one here. Okay, so let's say this is a plant cell. All right, this is a plant cell here. All right, good. Plant cell here like that. Okay, good. Okay, so the obvious thing about plant cell. Okay, so the obvious thing about plant cell. Okay, let's see. Let me bring this one in here. So I can write here. Okay. All right, good. So here now, so plant cells, eukaryotic cells. So let's talk about those obvious structures of plant cell. Like what is something unique about plant cell? Okay, let's start. Plant cell. Okay. So with this my diagram on the screen, okay, one obvious thing that plant cells tend to have is that plant cells, they have what? They have a cell wall. So they have cell wall. Okay. If you just say cell wall, I will mark you. I mean, basic level fine, but if you say cell wall, even this is cell wall made up of what? Made up of cellulose?
Cellulose.
You just hello, say please. What? Please, I didn't hear you.
I said cellulose cell wall.
Exactly. If you say cellulose cell wall, that's excellent one. Well done. Okay, but if you just told me cell wall, because bacteria, we just got to know that bacteria and then bacteria has a cell wall. Okay, so I expect that we should be more specific. So cellular cell wall is the best way to put it. Good. Okay, and then so we have the cell wall. Okay, so this cellular cell wall is the word. It's the outermost layer. Okay, so this cellular, okay, so let's make it cellular cell wall. That will make life easy. Okay, cellular cell wall. Okay, so cell wall made up of what? Cellulose. So cellular cell wall. So is that what? Is the outermost layer. Okay, is the outermost layer. And once it's the outermost layer, which means that it gives what? It gives fixed shape to the cell. Okay, it gives fixed shape to the cell. All right, it's the outermost layer, so it gives what? It gives fixed shape to the cell. Now, on top of that, on top of giving fixed shape to the cell, you can see that this cell wall is also fully permeable. Okay, fully permeable. Fully permeable. It's not like cell membrane. Cell membrane is selectively permeable. It doesn't allow so many things to pass through. But cell will allow almost everything to pass through. So it's fully permeable. And therefore, okay, it regulates the movement of substances into and out of the cell. Okay, into and out of the cell. Okay, so it regulates substances into and out of the cell. Okay, it regulates substances into and out of the cell. Okay, into and out of the cell. Okay, great. So it's regulate the movement of substances into and out of the cell. Okay, to regulate the movement of substances into and out of the cell. Okay, so that's cell wall for you. And when it comes to this cell wall, okay, we have two types of cell wall. All right, there are two types of wall. There are two types of wall. Two types. Okay, two types of cell wall. Okay, of this cellular cell wall. Okay, all right, and these are we have what something we call primary cell wall and secondary. Okay, secondary cell wall. Okay, so all right. Now, as the name implies, so let me. Okay, I don't want you to lose track of this one. So let me duplicate the slides. Okay, let me duplicate it. So okay, so here, agree. Now, the primary cell wall, okay, cell wall, it's found in all plant cells. Okay, and then the wall consists of what? The wall consists of mainly cellulose. Okay, there's another substance we call it hemicellulose. That's the primary cell wall, and a certain structure we call it pectin. You see, if you take a mango, have you seen mango? I think everybody has eaten mango before. Have you seen mango? If mango is not ripe, you can't really eat it. Okay. Now, when mango ripes, there is this enzyme in it, we call it pectinase. It is the pectinase that dissolve the pectin, so that makes it that substances that we can take it. Okay, so this pectin is like a gluey substance. It's outside this. So outside the cell here, we have this primary cell wall. Okay, so the cell wall outside this one is a primary cell wall, and this primary cell wall consists of cellulose, hemicellulose, and pectin. Okay, all right. The major component is cellulose. The major component is cellulose. Okay, and it is found in primary cell is found in all plant cells. Okay, all right. And the major component being cellulose, hemicellulose, and pectin. Okay, and then on top of that, it is found in the outer part of the cell. Okay, so apart from found in all cells, it's where it exists. It's where it exists in the outer part of the cell. Okay, it exists in the outer part. Okay, that's outermost. Okay, outermost part of the tree. All right, so that is the primary cell wall. Now let's talk a little bit about secondary cell wall. Now, when you talk of the secondary cell wall, okay, so let me duplicate the slide again. Okay, so that's okay. Okay, let me come here, duplicate the slide. Yep. Okay, so let me duplicate the slide. So now let's talk about secondary cell wall. So secondary cell wall. Okay. Now the secondary cell wall. I think I have to make a sketch for you. Okay, you still have this one in mind. Let me take this one off, and then let me draw. Okay, so that you can follow what I want. Now, if this is the plant cell like this, and this is the outer cell wall. Okay, what I'm bringing the outer cell. Now, the secondary cell wall goes like this. The secondary cell will go inside the primary cell wall like that. Okay, so something like this. So this one, so it's like this. All right, so and let's say this one becomes my cell membrane. So the first one here is the primary cell, which is the outer one. And the secondary cell wall is this, it grows, but it's not found in all plants, no? Okay, secondary cell is not found. So this is the secondary cell wall. Okay, you can see that it's thicker. It's bigger and thicker than the primary cell wall. Okay, all right, great. So with this secondary cell wall here, it's found, okay, within. Okay, so it's found within the word, found within in the inner parts. Okay, found in the inner parts of the primary. Okay, and then on top of that, it is where it is found in the inner parts of the primary. Okay, and then it is thicker. It is thicker. Okay, thicker, and then not found in all plant cells. Not found in all not found in all plant cells. So what are the main components? So the main components, so cellulose is there. It contains cellulose. Okay. I look at cellulose. Contains cellulose. It contains hemicellulose as well. And it contains something we call and then it contains something we call lignin. Lignin. Now you can see the lignin is absent in primary cells. So this lignin is it's a waxy substance. Waxy substance, okay, substance. All right, great. So this one is they are found. So this is impregnated with what lignin, okay. So they have cellulose, hemicellulose, and lignin. And the primary cell wall has cellulose, hemicellulose, and actin. Okay. Great. So please don't. And then, as I said, secondary cell is not found in all plants. It's only plant that thing to undergo secondary growth. Okay, that is where you find not all cells, like you find it in you find, if I say it's just secondary, I'm even wrong. Like let's say cells like so for instance cells like sclerenchyma cells, sclerenchyma, okay, cells, okay, these are great. These are those sclerenchyma cells. They are likely to have the lignin. Okay, but normal cells like parenchyma and collenchyma, they don't exhibit the word. They don't have the lignin. Okay, they don't have the secondary cell wall. So by the sclerenchyma cells, they have it. Okay, great. Not all plants have the word. Not all plants tend to have the secondary cell wall. Great. Any headache so far? So this is the cell wall bit, okay, of plant cell. Okay, all right. And then another last thing that maybe I should have said it in the first part here. Another thing that I should have said here was that cell wall is cell wall. It's not living. It's not living. Cell wall. Okay, it's not living. Okay, it's not living. Okay, good. It's not living. The structures there, they are dead. Okay, they are not really living. Okay, all right. So cell wall is not living part of the cell. Okay, you know the cell has living part and non-living part, and then the cell wall forms part of the non-living component of the cell. All right, good. So we've talked about the cell wall, okay, which is unique structure, giving the cell fixed shape, okay, and it's acting as the outermost layer, and I've elaborated more about that. Another obvious thing we can see from my screen here about the plant cell is that you also have what? Large permanent vacuum. Look at the vacuum. Large permanent centrally placed vacuum. Okay. So another thing, let me duplicate the slides again. Okay. So another obvious thing is that if you look here, you can see that they have what? They have large, okay, permanent vacuum. Okay, vacuum for what? For storing cells after photosynthesis. After photosynthesis, the cells are now reproduced. Okay, is stored here, and it is large and what? And permanent. So large and permanent what? Vacuum for storing cells up after photosynthesis. Okay, and don't forget that they are centrally placed. Okay, so large and permanent vacuum. And maybe another important thing that is unique to plant cell is the presence of what? The presence of chloroplast. Presence of chloroplast. Only plant cells, they have chloroplast. Okay, I think I have my diagram here. So if you look at the chloroplast, we'll talk about this in a little bit detail. But okay, so the chloroplast, you have this structure we call it the thylakoid. Okay, and the inside this thylakoid, that is where the green pigment is what is located for absorbing. So that's the green pigment. We're talking about the word, the chlorophyll. Okay, we're talking about the chlorophyll. All right. So we have the green pigments here. So the chloroplast is containing what? It's having the word, the green pigment, called what? Called chlorophyll. So for absorbing sunlight. absorbing UV for what? For photosynthesis. Synthesis. All right, great. So so this is that. So the obvious things we've talked about: presence of what? It's a presence of cellular cell wall. It has a fixed shape, and the reason it has fixed shape is that it has the cellular cell wall that is non-living, being the outermost layer of the what? Of the cell. Okay, and then it has large and permanent vacuum for storing cells up after photosynthesis. And then we can also talk about what? We can talk about one very prominent organelle that makes plants plant is the what? Is the chloroplasts. Okay, these chloroplasts, they contain what? The thylakoid, okay? This thylakoid. Okay, you can see this thylakoid, they are green, 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 green. And this green thylakoid, they are one having the what? The chlorophyll pigment, and they are capable of what? Okay, so they have something we call photosystems. Okay, we'll talk about them here maybe the next block when we are doing plant stuff. I'll talk about them. It's very easy, very simple stuff. So we have something we call photosystems within them, and these photosystems are capable of absorbing sunlight, and then for photosynthesis. Okay, and after photosynthesis, the starch granules are stored in the cell side. Okay, great. Okay, so these are what I could say about plant cell. Okay, the obvious things that I could say about plant cell. They have all the organelles as well. Okay, they have cell membrane. They also have the ribosomes. They have the mitochondria. They have the peroxisomes and nucleus and stuff like that. We shall talk about them other organ in a little bit detail. Okay, all right, great. So another thing about, so what are some of the examples of plant cells? What are some of the examples of plant cells? Okay, I think I gave you a few of them, but some of the examples of plants you can talk about the palisade. Okay, let's say, let me start with the obvious ones. Okay, so parenchyma cells. Okay, we can talk of parenchyma. Okay, good. Parenchyma. Parenchyma, I think it's double N. Okay, so then oh, I need something. Yep. And then we can talk of sclerenchyma cell. Okay, all right. And then we can even talk about, we have special type of parenchyma cell. Okay, then we have this special, we have the palisade. Palisade. So this palisade cell, they are found in what? In leaves, plant leaves. They contain a lot of what? So palisade cell is a special type of parenchyma. Okay, it's found in plant leaves. All right, contain a lot of chloroplasts. Okay, all right, good. So we can even take another special one we call the companion cell. Companion cell. This companion cell, it is found in that's a phloem. Okay, phloem. You know, remember phloem is what transport manufactured food to storage organs. Phloem vessel found in phloem vessel. Okay, companion cells. Okay, so all these ones are they are various types of plant cell. We can even talk about guard cell. That's also found in what? Leaves. Okay, leaves. Okay, so they were found in leaves. Okay, mostly under surface. The under, okay, maybe lower, let me say lower epidermis of leaves. What do? They close the word. They regulate the stomata. These are various forms of what plant cells that we talk about. Okay, this one, don't forget that this one is dead. This cell is dead at maturity. Maturity is dead. And don't forget that it is impregnated with what? With lignin. And this one provides strength. Strength, okay, mechanical strength. Right, mechanical strength.
Hello, sir.
Okay. Yes, please.
Yes, please. I would like to ask a question.
Please
Talking
go ahead.
about the companion cells.
Yes, please.
You only mentioned of the phloem cells, and I wanted to the phloem vessels of the phloem cells. I wanted to find out if the xylem cells also form part of the companion cells.
The xylem doesn't have companion cell. Okay. Well, let me
Okay.
let me let me let me let me explain to you why we need a companion cell. Just a minute. I will do that right now. Okay, so let me draw something for you to see here. For instance, you see when you look at the phloem looks like this. So for the phloem, there is this structure here. Okay, let's say something like this. Something like this. Okay. If you look at the phloem vessel, phloem is actually a tissue. Okay. So we have some structures here like this. Okay. So we have some structures here like this. Okay. Okay. Let me handle something like this. Let me put this one here. Okay. Now if you look this particular structure here. Okay. If you look at this phloem, this particular structure, we call this one sieve. We call this one sieve tube cell. Sieve tube cell. And there are holes here. Okay, holes here. Okay, the top here we call this one sieve plate. Sieve plate. Okay, holes here. So this through this hole that manufactured food will pass through to the storage organ. All right. Now you can see that this side, this here, this cell we call it sieve tube cell. This sieve tube cell is empty. It's like there's nothing in the cytoplasm, right?
Yes, please.
There's nothing in the cytoplasm. So how will it perform its metabolic activities? Okay, then God being so good, God has placed somebody on the side. So this particular cell on the side here is a companion cell. Okay, because this one lacks nucleus. It's almost like even cytoplasm because it's empty. And it is all that it is trying to do is to provide space so that the manufactured food can pass through. Okay, all right, good.
Okay.
So what happens is that because the sieve tube cell lacks the nucleus, okay, all right, and pretty much it should lack it so that it will enable it to transport the manufactured food, okay. So then on the side here, they've been given a companion. So this cell here, this companion cell here, okay, is the one that will, so all the metabolic activities such as growth, nutrients, and everything will all be performed by this one. So it depends on this one for its metabolic activities, just like the way we depend on our friends. So that is why it is called companion cell. Please, are you there?
Yes, please.
So because this one lacks those things, the nucleus, it lacks them. So we have this one here, okay, to help this one to carry out the metabolic activities. So all the metabolic activities of the C2 cell is controlled by the companion cell. Okay. All right, great. Any question on the plant cell that we've talked about? Okay, so I've given you examples of plant cells. We have the palisade, companion, guard. There are a lot of different ones. Okay, but these are the main ones that I've mentioned here. Then you have the collenchyma cells. Okay, collenchyma cells, they don't have lignin, no lignin, no lignin. Okay, they are living at maturity. They are not dead. Okay, the cells are living at maturity, and then they have an even thickness. Okay, they have an even thickness. Okay, an even thickness. That's one unique thing about them. So where you see companion cells where where flexibility and strength are required. Okay, so
Yeah.
the part. Hello.
Please, can you do us this favor by sending us this your annotated note. That
Yes,
note.
yeah, sure. Right after class, I'll do that. Yes.
Thank you,
Okay.
sir.
And you can see that I'm challenging you people. You can see that I can teach you that. Hello?
Yes, sir.
Hello? You see the way I'm teaching you people? You see,
Yes, sir.
you can see that I can teach you without book, right? Hello?
Yeah, we are listening, sir.
Yeah, so you can see that I can teach you without book. So I expect that when you also go to classroom, you should be able to master your concept. That you can also teach the students, okay, so well without book. Okay.
Thank you, sir. We are in.
All right, thank you. So I know you
Sir,
go and make me proud. Yes, go ahead.
please, where you wrote no lignin,
Yes,
it
please.
means the no will also affect living.
Oh, maybe the way
No
I wrote it. So I should put living.
living.
Okay.
Uh-huh.
Maybe then let me put the no
Then the
ligament.
on and then
Let me
uneven. Okay, if the no is there, it means it will be read no uneven. So that
so
have
let me
even.
put no ligament at the end here. At the end of
Okay.
the sentence. That will make sense. Okay,
Okay,
great.
so it means that the first part, which is living uneven thickness, they
Living,
are the
so
things
it starts, yes,
they
living
have
at
uneven thickness. Okay.
maturity.
Okay.
Okay, and then they have an even thickness, and then they have okay. So let me put have here have no ligament. Okay, good.
Yes.
That makes it a little bit easy for you to follow. All right, great. Okay,
And then
that
have
is
uneven thickness. Have uneven
yes.
thickness.
Okay, great. Have an even thickness. Good. All right, great. So these are various types of words. So types. So examples of word. So example, e.g. of the word of plant and cells. All right, good. Okay, great. So now that we've talked about this one, let's talk a little bit about animal cell. Okay, and then before we go to the organelles, okay, great. Now let's talk about animal cells. Okay, so let me say, okay, so let's see, animal cell. Let me go for blank page again. Let me take this one. Maybe this one will do. Okay, great. So, look at that. I've picked. Let's see. Okay, maybe this one will do. All right, great. So let's see. Let me go for animal cell diagram here. So that animal cell, animal cell.
This one, sorry for taking you back. Would like to find out if the bacteria you mentioned earlier on, in terms of the flagellum, some could
Mhm.
have more than one flagellum.
Of course, some could
Okay.
have it. Some
Okay.
have we done diversity already?
Yes, please.
Good. You see that I don't know whether you were touched that one, but there is this bacteria we call it
Dinoflagellate.
peritrichus. Peritrichus
And
bacteria.
say even dinoflagellate, the name dinoflagellate. They also have
Yes,
double.
yes. So they have to, okay. But dinoflagellates are not bacteria. They are different organisms altogether. They are algae. Okay, they are algae. Okay, that's the diatoms. Okay, all right. But we have bacteria that they will have a Leucotrochus. It didn't come across those names. Leucotrochus, Peritrochus, and okay. All right. So some yeah, obviously they have more. Some can have more. Some only have only one. Okay. Yes. So that's a good question. Well done. Okay. So let's see whether I can put my whether I can put a diagram here for you so that okay. So let's see. This is animal cell. Animal cell here. All right, great. So animal cell here, of course, that's nice. So one obvious thing we can see from animal cell, okay, is that okay. Let me carry this thing here. Let me show that I can put this. Let me take the layout and then so that to make life easy for me. Yeah. Okay, let me take this one. Let's see if I do something like this, okay, so that I can tell you. All right, great. So this is my animal cell on the side here, okay? And this animal cell here,
Great. All right, good. So this is my animal cell here like that. All right, with this animal cell, what can we do with the animal cell? Obviously, you can see that they lack what? They lack cell wall. Okay, they lack cell wall. And apart from the fact that they lack cell wall, okay, they also have what? You can see that, yeah, so they lack cell wall. That's the number one. Okay, so if you lack cell wall, it means that your shape is not going to be fixed. Okay, because so you have a cell membrane, so which means the cell membrane becomes your what? Becomes your outermost layer. And with the cell membrane, the outermost layer means that it will give variable shape the cell. Okay, so right now you can get two differences now: presence of cell wall, absence of cell wall. Plant cell has fixed shape. Animal cell has what? Variable shape. The shape keeps changing. Okay, and if you look here, if you look on the structure here, you can see that it lacks what? You can see so many organelles here, but there's one organelle missing. Okay, and the obvious organelle missing here is the word chlorophyll. So it lacks what? Lacks chloroplast. Okay, and so it is what? It is okay. So it is what? It can't what? Cannot perform with photosynthesis. Okay, and then if you look at the vacuum, now look at the vacuum here. Look at the vacuum here. You can see the vacuum is small. Okay, the vacuum is small. Look at that of the plant, large centrally placed vacuum. Here you can see this vacuum is what? So small in where? Temporal vacuum. Okay, temporal word vacuum. Okay, very small temporal vacuum. Okay, great. And then the vacuum, they are even of you can even get different types of it. You can get something like
Hello, sir.
Hello.
Hello, sir. I think
Please
today
go
is
ahead.
the day that I'll actually get the explanation to this. You have been teaching, I have been teaching, but of course I do question myself. With plants and animal cell, with respect to the vacuum, for the animal, for plants, we understand or we know that it is large, central, permanent vacuum. Then that of the animal, small and then temporal. Now my question is on the temporal. Temporal, in what sense? Does it mean that at times it will be there and then another time to go off? What does that mean? That word, temporal, what does that mean?
Okay, beautiful. Well done. That was exactly what I was coming to explain though. So we can get so the temporal in a sense that okay, if you take let's say food vacuum. Okay, let's say we can get various types of vacuum. So you can get food vacuum. Okay. The food vacuum will form when food comes into the cell. If you look here, I don't know whether you can see my screen very well, but let me enlarge this one here. Look at this one here. Look at this vacuum here. You see, if you look under my screen, you can see that, okay, I like this illustration. You can see that, look at a vacuum just formed, but look at how it formed. You see, there is this invagination. So the cell membrane pinched inward like that. Maybe a molecule came, right? A substance came, and then the cell membrane invaginated and then formed this vacuum. Okay, so it is temporary. It's not something is there forever. No, it forms as when it is needed. Now, in advanced organisms like us, we don't have vacuums in our cells, and you get to know it very soon. But if you take something like amoeba, paramids, those unicellular organisms, okay, especially those ones that tend to live in fresh water, they need vacuum. Okay, so I will explain it very soon. see. All right, I will elaborate all of them. Sure. Good question. So we have something like food vacuum. So the food vacuum forms when some big molecule is coming into the cell, if vacuum, some sort of vacuum can be formed. You can also get one vacuum, we call it contractor vacuum. This contractor vacuum, okay, mostly found in what? In freshwater protozoans. When I mean of freshwater protozoan, I'm talking about amoeba, about the word paramecium, okay, like that. Now these ones, you see, they are living in fresh water, so water will get into their cell by the process of what? Osmosis. And if water enters their cell by the process of osmosis, what they need to do is that they need to get rid of the excess water. So this contractile vacuum, what will happen is that the contractile vacuum can be full. Okay, so let's take it this way. Let me draw something for you here to see. So let's take it like this. Let's take a design, let me draw. Okay, great. Now let's take it. This is the amoeba cell like this, and this is the contractile vacuum. Okay, contractile vacuum. Okay, let me even draw it just like this. So the contractile vacuum, so amoeba is living in an environment that its concentration is higher than the water in the environment. So concentration of solute here is higher. So more solute here in the cytoplasm. Okay, solute. Okay, so more solute here. Let me say solute here. All right. Than outside the fresh water. So because of that, water will move into this cell by the process of osmosis. And when water moves in there, then the contractile vacuum will come, capture the excess water, and then when it becomes full, it will come and burst and release it out, release the content. So it will come to the surface, burst, and the new one will form. So this one also explains why it is temporal. Please, are we clear with that? So the contractile vacuum, you see the contractile vacuum here. What is the contractile vacuum doing? Contractile vacuum, okay, vacuum in what? In unicellular, unicellular organisms like amoeba, all right. Contractile vacuum in unicellular organisms like amoeba. What is it doing? It's controlling the water concentration. Okay, water concentration of the cell. Okay, and this process is called osmoregulation. Right. So what we do is that so that's why it is temporary. So here the water will come in. The water that comes in, the excess water will be captured by the contractile vacuum. And when the contractile vacuum becomes full, it will burst and release its content out. Okay, it will burst and release its content out. So that is why it's that very, very important. Okay, so it is temporary because it forms, burst, release, and a new one will form again. If it is food vacuum, if you look at my diagram here, you can see that let's say food particle is coming into the cell here like this. Okay, look at this one, and this one can go to form the vacuum here. All right. Okay, so this is food vacuum. Okay, and it is formed when it is needed. Okay, or when food substance comes into the cell. And then the contractile vacuum, it will capture the excess water, go to the cell surface, burst and release its content. Okay, and the new one will form. So making it temporary. Okay, so it is relatively small if you compare it to that of plants. So you can see this one is really. If you look at the size of the animal cell and the size of the vacuum, you can see that it is quite insignificant compared to that of plant cell. Okay, please, are we clear with that? Please, are you okay with what my explanation? Okay, so the one obvious thing is that a plant cell has large and permanent vacuum, where animal cell has relatively small and temporal vacuole. Okay, we have two types of those vacuoles. We have the food vacuole, we have the contractile vacuole. Okay, and the food vacuole for storage of food, contractile vacuole for performing osmoregulation. regulation. All right, good. Okay, any headache so far? And then maybe one last thing that I will add, okay, is the fact that animal cell, if you look at the living components of the cell, okay, if you take the animal cell, like the living components of the cell, the living components of the cell is what? Those living components, you see, water is not living. Water inside the cell is not a living component. Water, oxygen, carbon dioxide, ammonia, all those things inside the cell here, they are not living. Okay, but the living components are these organelles and stuff like that. Okay, the central, the nucleus, the mitochondria, chloroplast, all these are living structures. Okay, now those living components, the living components of the cell, we call it what? We call it protoplasm. Okay, so the protoplasm is a living component of the cell. The protoplasm is a living component of the cell. Okay, the protoplasm is a living component of the cell. Okay, great. All right. So now, if you compare the protoplasm, of what, of animals and that of plant cell, you see that the protoplasm here is denser. So this one is what? So relatively denser. It has denser protoplasm. So the living component, if you calculate the density of the living component of the protoplasm in plant cell and that of animal cell, you see that that of the animal cell is what? It's relatively denser. It's relatively denser. All right, good. So these are some of the key things about animal cell that I will talk about, like obvious stuff that of animal cell. So the lack cell wall, okay, you don't have to add cellulose to it. But if you add cellulose to it, it will sound ambiguous. It will look like maybe if you say they lack cellulose cell wall, if you write that, what it means is that maybe they have cell wall, but it's not made up of cellulose. So that makes it ambiguous. So please just so, and then they have variable shape. One of the reasons they have variable shape is because they lack cell wall. Okay, they lack chloroplast, and then their vacuole is small and temporary. And then on top of that, their living component is relatively denser than that of what? That of plants. So if I ask you to tabulate 5 differences between plant cell and animal cell, I know you can do that for me beautifully. Okay, good. So nice. Okay, good. Now, so the next thing I'm going to talk about, okay, then after that, I'll give you a break. Yes, a question for me, please go ahead.
We know that the protoplasm is made up of the nucleus and the cytoplasm.
Not really. Not
Okay.
really. Protoplasm is actually the living component of the cell. Not necessarily, because inside the cytoplasm, you can get water, and water is not part of protoplasm. Water is not living.
Okay.
We get it. Yes. Water and carbon dioxide and oxygen, all those we can call them cytoplasmic inclusions. Okay, we can call them cytoplasmic inclusions. Okay, so all right. So let's say okay. Well, okay. Let me go. Let's see.
Okay. So with this, it means the only living organelle in the animal and the plant cell is the nucleus. Is that so?
No. So all the organelles are living. So let's see. Okay. Let me go step by step. I will touch on what you are asking. I'll
Okay.
come there very soon. Okay.
Thank you, sir.
So let's say all cells have cell membrane. Okay. They have DNA. I think to bracket genetic material. Okay, material. And then they also have what? Ribosomes. Ribosomes. So this one is all cells must have this. All cells, no matter who you are, you should have this one. Okay, great. Now, so and these, you see, outside these DNA and ribosomes, they dwell within the what? They dwell within the cytoplasm. Okay, so all right, so they dwell within the what? The cytoplasm. Okay, so if I go to the cytoplasm, if I write about the cytoplasm, okay, which side? The cytoplasm is like almost everything inside here. Okay, the cytoplasm is everything inside here. That's the cytoplasm. Now, with the cytoplasm, there are things there. So we have two things there. We have the living component. We have the living component. Living component. A living component. And we have the non-living component. Non-living component. Okay, so we have the living component. Okay, and non-living component. Okay, so you have living component and non-living component. All right. Now, for the living component, we can talk about what? The organelles. So organelles are the living components. So organelles are living components. Okay, and non-living components, yeah, you call them cytoplasmic inclusions. Okay, and that is, we can talk of PG, we can talk of water, oxygen, CO2, we can talk of sometimes even yeah, like water, oxygen. Yeah.
But sir, please, we understand organelles to be the structures found within the cell.
Some
So I guess the organelles are the structures found within the cell.
come again.
Cell wall is not, please, is cell wall an organelle? Cell wall.
It's not an organelle.
So I understand organelles to be structures found within the cell. So now that you are talking about the non-living part, I think the cell wall has to be included.
No, what I'm saying, you see, I started with cytoplasm. Is that not it?
Sir, can I come in tomorrow?
Yes, come in.
The cell wall is not an organelle. The organelles are found, all those components that are found in the cytoplasm are said to be the organelle, if I'm right.
Yeah, but not all of them. So those structures within the cytoplasm. So within the cytoplasm, we can get living structures, and then we can get non-living structures. So the non-living ones are what we are terming as the cytoplasmic inclusion, like water, carbon dioxide, oxygen. Okay, those ones they are found in the cytoplasm, but they are not living. Okay, so we call them cytoplasmic inclusions. And the living structures, we are actually, it's more of exclusively. Okay, in fact, we can. It's not. This is not the only living structure we can get from there. In there, we can get amino acid. We can get amino acid, and amino acid are associated with living. Okay, amino acid is not living. Amino acid is living. They are organic. The organic structures. Okay, all right, good. So
what
but we
about
have
sugar? Sugar.
We can put sugar there as well. Okay, they are organic, not living as okay. Well, let me put it organic structures. Let me put it organic. Okay, organic. Maybe.
So we are still living and they have life in them.
You see, I'm coming. So let's say this is an organic structure. They are associated. They are directly associated with living. They come out of living. Do you understand that? but water and oxygen and carbon dioxide, they are inorganic substances. So that's so those ones are cytoplasmic in food. But if you take the organelles are unique structures. Okay, so it's like more or less little organs. The word organelle simply means little organs. Okay, in Latin. Okay, so how do we define the organelle in this context? So the organelles. Okay, so for the organelles, they are what they are. So under the living components. Okay, under the living structures. Okay, so I will start by saying they are the living structures of the cytoplasm. Okay, that they are living structures of the cytoplasm that carry out what that perform
Specific functions.
specific functions within the cell. Okay. So under this one, okay, yes, they are living, but they are performing some sort of specific, they are quite organized structures, okay, within the cells, and then they perform specific function. All right. Now, so under even these organelles, we can get two types. We have membranous, let me start with non-membranous, so under the organelles. So two main types of organelles are, so we can talk of what two main types of organelles are. We have membranous organelles, and we have what? And we have non-membranous organelles. Okay, we have membranous organelles and non-membranous organelles. Okay, so for the membranous organelles, okay, when we talk of membranous organelles, that's where we are talking about the nucleus. Okay, I think I've mentioned them. Endoplasmic reticulum, Golgi body, peroxisomes, mitochondria, chloroplasts,
Lysosome.
Okay, so these ones you can see they are bounded by membranes. Okay, these are bounded by membranes. So under this one, so these are bounded by membranes, and then the non-membranous organelles we can talk of ribosome. These ones they don't have membrane bound. You see, so in SS textbooks, there's definition in the SS textbooks that I've been complaining about, and so it has not been changed. So I always tell people that they have to be careful when they are reading. This is, you see, they define organelles as they are structures within the cell that are bounded by membranes. But if you say that, it means that you are making, you are saying that ribosome is also bounded by membrane, but that's not true. So what they should say in that SS table, they should add that structures within the word, let's see, the living structures of the cytoplasm, okay, that may be bounded by membranes. For biology guys, SS biology guy, the mistakes in it, they've still not corrected them for so long. I don't know why. Okay, that may be bounded by membranes, okay, and perform specific function. You see, so why? Because if you say there are structures that are bounded by membrane, that's not entirely true, because ribosomes are not bounded by membrane, the organelles, not bounded by membranes. Another one is centrioles. Centrioles are also not bounded by membranes. Another one are the cytoskeletons. Cytoskeleton. Okay, cytoskeletons. In cytoskeleton, we have three types. Okay, now let me put G here. Okay, EG here. Cytoskeleton, we have three types. We have Mike, we have microfilaments. We have intermediate filaments. And then the last one is microtubules. The microtubules are bigger. Okay, so microtubules are bigger than intermediate. Intermediate is bigger than microfilament. All right, so these are the organelles stuff. Okay, any headache so far? So please, when you are teaching at SS, please make sure the definition. Don't say that their organelles are bounded by membrane, just like the lobe. Not all of them. You can get ribosomes, centrioles, cytoskeletons. They are all organelles. They are all living structures within the cell, but they are not bounded by membranes. Okay, but they perform specific function. Okay, all right, good. So these are the two main classes. So we have the membranous organelles. These ones are bounded by membranes, and we have the non-membranous organelles. They don't have membranes. The ribosome, the centrioles, and the cytoskeleton. These are non-membranous organelles. It's very clear with that. Okay. So based on this one, now we can take them one by one and talk about them. Okay. Good. So this is what I have here. Okay, great. So I've explained this one already. So we're going to take them one by one. But before that, I think what we'll be doing is that we'll talk about cell membrane. Okay, after the cell membrane, then we'll talk about the organelles one by one. Okay, all right. So maybe 8 minutes time we'll go on break. Okay, so let's talk about cell membrane. Okay.
Hello,
Okay.
sir.
Yes, please.
Sir,
Yes,
please kindly permit me to ask this question. It's not part of the studies though, but I want to satisfy my curiosity station in my mind. Read those online. Having you, please. Oh, are you the coordinator for this course?
I
The
am.
coordinator for this course. Oh,
I am the
okay.
coordinator.
Okay.
All the
Okay.
courses that I'm teaching, I'm the coordinator.
Okay. My reason is with respect to the areas where questions will be set to cover. In a sense that you see there are a lot of links that were sent, and I happened to join one. After listening to the voice, I realized it wasn't you, so I left. And upon my realization, it's like they are far. They've gone with respect to the slide. They are very far from us. So I was contemplating that. So in that sense, how are the areas for the questions to be covered going to be? And
Don't
we love
worry.
you. I in person. I really love the way you move. I really love you. In fact, you have really started something within me, and I'm loving it. But my concern is those ahead of us, and we to hear how the questions will be in order not to have any problem at the end. That's the reason.
Don't worry. Okay. My interest is even if we will do small and you understand it, okay, I'm okay with that. Okay, don't worry. I'm your coordinator, so I will take the final shot. So don't worry. Okay, all right.
Okay.
It doesn't matter how they run. I'll be the one who will be setting the questions and everything. Okay, so don't worry. Don't worry. Okay.
God bless
Thank
you,
you.
sir. God bless you, sir. God bless you, sir.
Why are the men happy? Hey, please, don't be happy. You've not seen my questions
So
yet.
please, you're
We are just now learning the method you are using. We love the method.
You've not seen my questions yet too. My students usually they laugh in class, but when they get my questions, sometimes they cry. So please.
First is we are for our body.
The two of us I thought that will not be easy. That one I know that it will not be easy, but I just wanted to be fully assured that with you.
Now the men are scared. Now the men are scared. Okay, don't worry, you'll be fine. You will be fine. Okay. Hey, you question, why are you laughing? Don't worry, you'll be fine. Mercy and Co are here. Mary and Co are here. My wonderful ladies are here. Helen is here. And so please, the question will be very easy. Don't let me say anything. Don't you know my ladies are here? Okay. I have to make the question very easy for them. Okay. All right. Okay, so please don't worry. We'll finish. We'll finish everything. Okay. Even if you don't finish it, I will make time so that we'll finish. Because these aspects, the biology, the central thing about biology, cell is the central thing about biology. And so once you miss it, but trying to connect it up at some part becomes very difficult. And we know what is in SSBs and stuff like that. So we try as much as possible just to break it down very well so that you won't have any issue when you stand in front of the class. Okay. The same way I'm teaching you, I expect that when you go to class, you also do likewise, so that I'll be so proud. Hear that, hey, my former student is doing marvelously well. I'll be so proud and I'll be so happy. So I know you'll be making me proud. So let's move in the normal pace and then, but we'll finish. Don't worry, we'll finish everything. Don't worry. Okay. All right. Good. So we are talking about what? We are talking about the cell membrane. Okay. Very good. So the cell membrane here. Okay. We know that the cell membrane, it is what? It is enclosing. Okay, enclosing. All right. You observe this picture first. Okay. After you observe this picture, I'll take a blank page and I'll talk about something. Look at this picture here. Now, if you look at this picture here, you can see more or less two layers. Okay, you can see some two layers here, right? And then, so these two layers like that, okay, so two lipid layers. Okay, so because of the two lipid layers, you can see bi means two, so bilayer. Okay, so look at that here, look at this one here like that. Okay, and then you look at the protein. Some of the protein have some carbohydrate attached to them. Okay, so I want you to observe this one before I move on to the next one. Okay, I'll take a blank page and do something. Okay, great. So please, have you observed this one well? You can see two layers, right? Two layers. Okay, we can see some protein. Some proteins are just hanging on top here. Some are going through the entire length and stuff like that. Okay, good. Excellent. So now let's see something here. All right. So there is this particular molecule. Okay, let me draw it for you. This molecule is here like this. Okay, this is a very beautiful molecule here. All right, good. And this molecule, there is this molecule is what? This molecule is called, so this particular, this head, this one, so here like that. Okay, so this is the tail. So let me call this one the tail. And this tail here doesn't like water, just like most men. Okay, most men don't like to bath. Okay, all right, good. So the tail doesn't like water, and so when you don't like water, the term for you is called hydrophobic. Okay, phobic. You know, phobic means heat. Okay, hydrophobic. And then this is the head region. This head region loves water, so it is what? Hydrophilic. Okay, good. Now, when a molecule has this characteristic, whereby some part of the molecule loves water, some part, so more or less this part here, I don't know whether you've done this one in chemistry, is polar. The head region is polar, right? Okay, it loves water, is polar. And this tail region it's basically lipid. It's lipid. Okay, and this side is what? It's non-polar. Non-polar. Okay, good. Now, the reason this head region is hydrophilic or is polar is because it contains something we call phosphate. And remember, phosphate is what? PO4 minus what? 3 like that. It contains phosphate. So this phosphate is water loving. So what does it mean? So there's this molecule. This molecule, so there's scientific term for this molecule. This scientific term for this molecule is called what? Let me see. Let's see what I can. It's the so that will, okay, great. So this molecule, why? Okay, so my drawing is here like this. So this molecule, okay, molecules that have both polar and non-polar parts is called what? It's called both polar and non-polar parts is called amphipathic molecule. Amphipathic molecule. Okay. So this particular molecule, what is the name of this molecule here? This molecule is called what? So it has, we have the polar head, it contains phosphate, and it has a lipid. Okay, so because the phosphate, okay, here head, it makes the head love water, and the tail doesn't like water, so the tail stays away from water, so it is hydrophobic. And a molecule with this structure, having polar and non-polar parts, is scientifically known as amphipathic molecule. Okay, and this molecule we are talking about here, this molecule that we are talking about here like that, okay, this one that I've drawn here, okay, this molecule that I've drawn here, it is called what? It is called phospholipid. A phospholipid. So we know that this phospholipid, because it contains phosphate head, which contains what? Which contains phosphate, and the tail is the lipid, which is the oil part, which doesn't like water. So this particular structure is called what? This molecule with this character is called amphipathic molecule. Had a hydrophilic head and hydrophobic tail. Now, if you research has shown that if you put this molecule together, so let me draw it again. Okay, let me draw it again. If you put this molecule together like this, okay, in a container, and you leave them, it will go this way. They will rearrange themselves like that. They will rearrange themselves like that. All right, good. So when they do the rearrangement, so you see, they will rearrange such a way that the head region will be pointing outside towards water. So this will be towards what? Water. Towards water. This side will also be towards what? Towards water. So that is it. So the head region will be pointing outside towards water. Okay. And then the tail region will be pointing inside, like that, away from water. So head region will be pointing outside towards water, and then the tail region will be pointing inside. Okay, will be pointing inside. Okay, away from what? Water. And so this rearrangement is what you call bilipid layer. Okay, all right. It's a bilipid layer. But which bilipid as well? What sort of lipid are we talking about? The lipid is called phospholipid. And so if you want me to put term into this one, what will I call it? I'll call it what? So I'll call this layer thing, okay, I'll call this layer thing, I'll call it what? Phospholipid, phospholipid bilayer. Okay, phospholipid what? I'll call it phospholipid bilayer. So if you look at my drawing here, these two layers are forming what? Phospholipid what? Bilayer. Okay, good. So back in 1972, people were finding it difficult to describe the cell membrane. Okay, so two scientists, okay, two American biochemists by name, we have Sanger and Sanger, I miss memory. Sanger, Jonathan Sanger, and hey, how can this thing disappear from my head? Two American scientists. No, it's not even Sanger. It's Singer and okay. Well, let me explain. The name will come. Don't worry, it will come very soon. So these two, what they said was that these two layers here, we have two phospholipid layer, okay, and these two phospholipid layer, okay, they are interspersed with what? With protein, okay, they are interspersed with what? With protein. All right, is that not it? Okay, all right. So we have this. When they have the phospholipid layer forming the cell membrane, okay, so this layer will have inside of it, okay, inside of it, we have proteins inside of it, all right, and this protein inside of it is such a way that is giving this sort of some dynamism, okay, dynamism, all right, some dynamism. So you see protein hiding inside of it. You have other lipids there. We have different type of words. Let's say different type of molecules, all manner of molecules. Okay. So people started coming with different models to describe the cell membrane. Okay. So let me copy this and paste it here. Okay, so people are coming with what? With different models to describe the cell membrane. So these two American scientists, Singer and Nicholson. Please double check it for me, okay? All right, double check it for me. Okay, double check it for me. We have Nicholson, yeah, Nicholson. Okay, so we have the Singer and Nicholson, okay? I think Jonathan, J. Singer, Jonathan Singer, and Gath, okay? Gath, that's G, Gath. The Gath is this, okay? Gath Nicholson, yeah. Okay, sometimes the names can escape me sometimes. All right, so we have these two people, okay? So these two people, where people were coming with different models, okay? All right, coming with different models, like the way we have the globe being the model of the earth. Okay, people are coming with different models to describe the cell membrane. There are various ones, but the most acceptable one. Oh, the names didn't come here. Okay, oh, do you know what? Let me take one online for you. Okay, so cell membrane. Okay, let me take the one with the labels and stuff like that. Okay, all right, great. So copy. Okay, great. Okay, this drawing tool is nice. So let me put it somewhere here and then bring this one. So, okay, great. So Jonathan. Okay, Jonathan. Jonathan and where? Jonathan Singer and Gath Nicholson. Okay. like that. Okay, great. Okay, great. So these two people came out with a model, something to describe the cell membrane, how the cell membrane looks like. How does it look like? So they said that this cell membrane, okay, the model is something you are using it to describe something like the way you have globe being the model of the earth. Okay, so that when I look at the globe, I can look at the maps and stuff like that. Okay, good. So these people came up with a certain model, which we call it fluid mosaic model. A fluid mosaic model. All right, and this fluid mosaic model is basically saying that the cell membrane, okay, we have the phospholipid bilayer. Okay, you know the phospholipid bilayer, you can see the head region here, and then the head region is pointing out and out like that in the outer part. Okay, let me use this one too for you here. Okay, so let me shape it a little bit. Okay, so put this side. Okay, good. So if you look at the head region, okay, if you look at the head region, you can see that the head region is here, it's pointing outer, like that. So outer, 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 outer. And in between, the interspace within them is the word, are proteins, okay? Different, all manners of proteins are there, all right? Some proteins are going, they are within, others are what? Others are just hanging in here, others are going through the entire length of the word, of the cell membrane. So this model whereby we have the word, the phospholipid word, lipid phospholipid bilayer, okay, interspaced with what? Interspaced with proteins, okay, is what we describe as the word, the fluid mosaic theory. The fluid mosaic theory. So this fluid mosaic model, okay, this fluid mosaic model is just saying that we have the word, we have the phospholipid bilayer interspaced with what? Protein. And it is these two American cell biochemists, okay, they discovered it in what? In 1972. 1972. All right, great. So J. Singer and G. Nicholson. Okay, so the two of them gave out the word, the fluid mosaic theory, saying that we have the cell membrane interspaced by what? We have phospholipid bilayer interspaced by proteins. Okay, now look, if you look at this diagram I have on the board, you can see we have two different types of protein in here. Okay, you can see some of the proteins are what? They are just, look at this protein. This protein they are hanging on the surface. Look at this protein. Now those proteins hanging, okay, proteins on the surface, okay, the proteins on the surface. Okay, so all these proteins, we can call them, let me call membrane proteins. Let me be specific. Membrane proteins. These are membrane proteins. So the membrane proteins that are what? The membrane proteins that are that are on the surface. On the surface, okay, on the surface of the are called what? Are called peripheral. Are called peripheral protein. They are called peripheral protein. Okay, and then the words, the ones that are embedded within the cell membrane, you can see these ones are embedded. These ones are within the cell membrane. See, these ones are just hanging on the surface. Look at that. These are on the surface. These ones are within. So the ones that are within the word. Okay. So the ones that are within the cell membrane are called. So the within, okay, within the word, the cell membrane, cell word membrane cell membrane, they are called what? Integra. Integra protein. Okay, and even the integral protein, we can get two types. Okay, you see the integral protein, some goes through the entire length. Some go through the what? They go through the entire length they span. Okay, so here, let me straighten this in a little bit. Okay, I think this one can go. I think it's done with the job. Okay, so that I can have more space to write. Okay, so let me put this one here so that I can write. Okay, so let me bring the diagram here like that. Okay, so the one that tends to go through the word, the entire length. Okay, so within the word, within the cell membrane, we are talking about what? We are talking about within the cell membrane, we can get two types of protein. Okay, so there are two main types of protein there. We have the peripheral, which is just on the surface, like the one here, and then we have integral within the cell membrane. Now, with this integral, we can get some types of integral protein. They span. Okay, the integral proteins that span across the entire length of the word, the integral that span across the entire length of the cell membrane. Okay, it's called what? Those ones are called transmembrane proteins. Okay, so this one, like this channel protein here, is a transmembrane protein because you can see that it goes through the entire length. Okay, it goes through the entire length of the word of the cell membrane, and so those ones like that, they are called what? So they are specific type of integral protein called what? Transmembrane protein. Okay, all right. Any headache so far? So we are describing the cell membrane. In the cell membrane, we are talking about what? The major components of the cell membrane, we are talking about the phospholipid. And this phospholipid is having this bilayer. So we describe the phospholipid bilayer. And this phospholipid bilayer are interspersed with what? With proteins. Some of the proteins are just on the surface. This we call it peripheral protein. Some are embedded within. These are integral protein. Some of the integral proteins spans across the entire length, and those ones are called what? They are called transmembrane proteins. Okay, so the whole of this structure, which we are describing as a fluid mosaic model, was brought by these two American cell biochemists. Okay, we call it Jonathan Singer, okay, and Gat Nicholson in 1972. Okay, so the two of them actually brought the fluid mosaic model to describe the cell membrane. And that simply means cell membrane consists of what? Phospholipid bilayer interspersed with proteins. Okay. Please, any questions so far?
Yes, sir. Please, can you write the phospholytic bilayer in full, especially the phospholytic? So that in case, yeah, I mean lipid. Yeah. Lipid, sorry. Lipid.
Phospholipid bilayer, okay, interspersed with protein. So this model, okay, and right now this is the most accepted model of the cell membrane, okay, and given by what? J. Singer and J. Nicholson in 1972. Great. So we'll talk briefly about each component of the cell membrane. Okay. Now we just said that, you see, so we have this this lipid. Okay, so phospholipids here, which is the major component. And you know what? As a matter of fact, this phospholipid, let me duplicate my slide. Okay. Let me duplicate the slide a little bit. Okay. As a matter of fact, this phospholipid that we have here, okay, this phospholipid that we have here, Depending on it, when you come to lipids, we can group them into two. I don't know whether you've done it in organic chemistry. We have saturated fatty acid and unsaturated fatty acid. I don't know whether you remember something like that. Okay, so depending on where you are on the planet, depending on where you are, so those of us who are in the tropics, okay, so this our leg here, this leg we have here, this leg is the lipid part, and this is the phosphate part. Now this lipid part here sometimes is the fatty, this is the fatty acid, the lipid part is the fatty acid. Now, depending on where you are on the planet, this phospholipid can be what? This phospholipid, the lipid part, the lipid can be saturated or unsaturated. Saturated means that it contains single bonds throughout. All the carbons they're having single bonds throughout. Unsaturated means that so unsaturated means at least one double bond. One double bond. Okay, so I'm using these two lines to indicate double. Let me enlarge this one a little bit. So at least one double bond, okay? At least one double bond. It's one double bond. Okay, good. So saturated, at least one double bond is there. Okay, good. So that's it. So now depending on where you are on the planet, you see, let me give you an example of saturated and unsaturated. You see, when you come to palm oil, you see palm oil, if you put palm oil there, we have a type of palm oil, especially the agric one. The agric palm oil from those agric palm, most of the time when you put it there, they tend to solidify quicker. And then, but if you put some of the local one, it doesn't solidify quicker. Okay. Now, most of the time, those oils that tend to solidify quicker, they are saturated. Okay, they are saturated. And so those of us in the tropics, okay, they're saturated. Those of us in the hot part of the world, usually our cell membrane has saturated fatty acids. So those in what? Warm climates. So warm climate will have what? Will have saturated. So cell membrane in warm climate will have the saturated ones. And then the one that doesn't solidify quicker. The one that doesn't solidify quicker. The one that doesn't solidify quicker, they are the ones in where? The ones that doesn't solidify quicker, they are the ones in the ones that doesn't solidify quicker. Okay, they are the unsaturated one. Those doesn't really solidify. They are the unsaturated ones, and they are mostly found in where? Those organisms living in where? In temperate. Okay, those temperate climate will have more of those ones in their cell membrane. Okay, because it doesn't. So here it doesn't really solidify. So if you see, if it freezes quicker, it means that in no time, the people in Alaska and Russia, they will not be in existence, but the cell membrane will freeze. Okay. Now, but so most people they have what? Their cell membrane has high level of what? Unsaturated hydrocarbons, okay, in their phospholipid. And we have, those of us in warm weather here, we have the saturated ones, okay? So these are things you should bear it in mind. Now, within the cell membrane, apart from this phospholipid, there's another lipid there. There's another lipid that we call it what? We call it cholesterol. Okay, cholesterol. Okay, cholesterol is very popular. Okay, it's very, very popular in recent times. Okay, very, very popular word now. Okay, So cholesterol like this, okay, when you come to this cholesterol, okay, as I said, that is quite popular in recent times. Now, for the cholesterol, what is cholesterol going to do? We have this cholesterol in the cell membrane, okay? So let me show you something. Let me draw so that you see what I'm talking about. Now, if you go to the, let me use different color here. Okay, so let me pick red. Now, if the cell membrane is here, you will get cholesterol like this, hiding here. Okay, sometimes they will go through the whole length like that. Okay, they will go like this. They will be like this. Okay, now what is this cholesterol doing? Now, what this cholesterol is doing is that it is doing something we call we call it fluid buffering. Okay, so fluid, it regulates fluid buffering. I will explain. It's not difficult. What it does is that this particular one is very strange. This cholesterol in our cell membrane is very, very strange. When the weather is cold, you see, when the weather is cold, the phospholipid, they are likely to get stuck to each other, right? Is that not it? When the weather is cold, okay, this phospholipid here, they are likely to get stuck to each other because they will be slow in moving. All right? Is that not it? Are you with me? When in very cold weather, this cold weather, molecules tend to move slower in cold condition. When temperature is low, they do what? The molecules tend to move slower. So they are likely, these phospholipids are likely to get stuck to each other. And if they are even saturated, then it even worsen the case. So what this cholesterol here, what this cholesterol is doing here, this cholesterol doing here, this cholesterol is doing here is that it ensures something we call fluidity buffering. Okay, fluidity buffering. Fluidity, okay, fluidity buffering, meaning that when the temperature is cold, okay, then this cholesterol in our cell membrane will become more fluid, so that it will cause the phospholipid to move faster. Because when the weather is cold, the phospholipids are likely to get stuck to each other. So what happens is that when the weather is cold, this cholesterol in our cell membrane, instead of becoming solid, it doesn't become solid. It becomes more fluid. And when it becomes more fluid, it causes the phospholipid to agitate so that they can move faster, so that they don't get stuck to each other. And on the opposite side, when the weather is wet, when the weather is also hot, you see, when the weather is hot, the phospholipid will be moving too fast, and it is dangerous. When the phospholipid moves too fast, remember the cell membrane acts as what? Selective permeable membrane. And this gap between them here must be maintained like that. Okay, so only few substances can walk through. Only few, not everybody can go through. Okay, maybe oxygen can squeeze through like that. Maybe oxygen here can squeeze through like that here, like this. Maybe carbon dioxide can squeeze through out like this. But other substances shouldn't go through. Now, if these molecules are moving too fast, if they move too fast, if you are not lucky, the space between one phospholipid and another phospholipid here will be big. And so molecule that shouldn't cross will begin to cross. And that will be a disaster when the temperature is too high. When the temperature is too high, the molecules will be moving too fast. So when temperature is too high, then this one will rather what? This one will become what? The cholesterol will begin to solidify. Okay, so when the temperature is too high, okay, high temperature, the cholesterol will begin to solidify so that it will slow the movement of the phospholipids. Okay, so please have you seen what it's doing? So it's like when it just like so it is buffering, like the way we train kids. We are buffering them. When they do something nice, we praise them. If they do something bad, we penalize them. So basically that's what happens. When the molecules are moving, when the phospholipids are moving too fast, when temperature is high, then the cholesterol will solidify a little bit so that it will slow down the pace it moves. The phospholipid will move. And again, when the temperature is too cold, it's cold, and then the phospholipid are getting stuck to each other, then the cholesterol becomes a little bit fluid, so that to increase the agitation of the phospholipid. So what this phospholipid in our cell membrane is doing is that it actually regulates fluidity buffering. Okay, fluidity buffering across what? Fluidity buffering across physiological temperature. Fluidity buffering across physiological temperature. Okay, so what this cholesterol is doing is that to ensure what? Ensures regular fluidity buffering across physiological temperature. All right, so that is, so we've talked about phospholipid in here. Another lipid in there is the cholesterol. Okay, so the cholesterol, what is it doing? It's actually regulates fluidity buffering across what? Across physiological temperature. Okay, that when it is too cold, when it is too cold, the cholesterol becomes much fluid, so that it will increase the agitation for the phospholipids, so that they don't get stuck to each other. And when it is too warm, when it's very warm, this one will rather solidify, slow down the pace at which they move. Okay, good. Any headache so far? Any headache so far? Okay, any headache so far?
Yeah,
Any headache
yes, sir.
so far?
Please, the bullet you placed under across the physiological. If you
It
can,
was going
if
here,
you
it was going to this side. It was not making this side visible. That's why I bulleted it down.
okay, so
It's
if you can take
continuing.
it off so that we'll know it's a continuation. Else we'll consider it as a point. Yes.
All right,
Thank you.
great.
Thank you.
So this is that here, so that so it regulates the what? It regulates fluidity buffering across physiological temperatures. Okay, all right, good. Now another important thing that is inside this cell membrane is the what? Is a membrane protein. Okay, so let's talk about the membrane protein. Okay, so look at that. I have this beautiful diagram here. Okay, good. So this is okay. So that's beautiful diagram here. So this big one like that can be termed as this particular blue one like that. Okay, so this this is a protein going through the entire length like this. So we can, this is integral protein, but the type of integral protein here can be called what? Transmembrane, because it's going through the entire length. Okay, all right, good. And then okay, so let me finish this one, because this one is already in your notes, so let me finish it. So when it comes to, let's come to the membrane protein. So membrane proteins, okay, proteins. So the membrane proteins, as I said earlier, we have two types. We have the peripheral and what? And integral. And some of the integral will go through the entire length, and those ones are called transmembrane. Okay, transmembrane. Okay, transmembrane. Okay, transmembrane protein. Okay, great. Now these membrane proteins, okay, let's see some specific ones like that. Okay, let's talk about them in general. What are their functions? What are they doing? I asked this question last year about functions of plasma membrane, plasma proteins, and some people are saying that for providing energy, repair growth. You see, this is something specific. So please make sure you take note of it. It's something specific to the cell membrane. So you can't say growth and repair of worn out tissues. No. That's not what we want. Okay, great. So this is membrane protein. So clearly they didn't know what I was talking about. So if you look at the membrane protein, what are the functions of membrane protein? Okay, so let's see one. Okay, one. Okay, so functions of what? Membrane proteins. Okay, so let's say one. You can talk of what? You can talk of cell identity and cell recognition. Okay, great. Now, when you talk of cell identity and cell recognition, you see on your right now, if I remove your, let's take it that when people are going for transplants, okay, first thing we talk about is that it's one of the things we talk about. Let me even bring the picture here as well. Okay, when people are going for transplants, okay, one of the things we look for is that we want a situation that the things on the surface here, that these things, okay, this glycoprotein, especially the surface protein here, those so we have on this surface here, we have something called glycoproteins, okay, glycoproteins. Glycoproteins, right? And these glycoproteins, what do? They give cell its unique identity. All right, they give cell its unique identity. So you see, these proteins connected, these are carbohydrates connected to proteins, forming glycoproteins. So they give cell its unique identity. So during organ transplant, okay, what happens is that we need to match what is on the surface of your cell membrane. So that is why ideally we expect that during organ transplant, we expect it from what? From to pick the donor, the one who is going to give you the organ, who is donating organ, should be maybe the closest relative as possible. So ideally, organ transplant like this, you should take from your mother, your father, or maybe a child, or your sister, or your brother. Okay, so that the glycoprotein here will be as similar as possible. Okay, so because of this cell uniqueness, so for instance, if let's say a pathogen enters my body, this pathogen will have its own glycoproteins on the surface like this. And so these glycoproteins, my white blood cells will come and say, like these glycoproteins, are they the same as the one I have in my body? No. So if they are different, then they'll come and attack the cell. Okay, so these glycoproteins and such like, they form something we call antigens. Okay, they form the what? The antigens. Antigens on the surface of the what? On the surface of the cells. And so the antigens are used to detect. They are being used for the white blood cells to detect whether the cell belongs to the organism or it is outside. Okay, so when they come in, when they say any pathogen, let's say amoeba has entered my body, what is on the surface of amoeba will help my white blood cell know that, oh no, this amoeba, this anti-amoeba that has entered my body, it's a foreign material, and so you should attack it. Okay, good. So the one thing about the cell protein or membrane protein, specifically the glycoproteins, they give what? Cell to cell recognition. Okay, cell to cell recognition, or cell wide identity, especially the glycoproteins. So for instance, you see that if you take something like the blood groups, okay, you take something like the blood groups, okay, the blood groups like that. So you see blood group, let's say, all right, let me take different color. Now, there are some people who have blood group A, so this one will have glycoprotein A on the surface of their red blood cell. Some will have blood group B, they have B here. Some have A, B, one here, one there. So A is here, B is there. And some doesn't have anything. So this is blood group A, blood group B, blood group AB, and blood group O. So me being blood group O, I lack what? I lack that glycoprotein, that specific glycoprotein on the surface of the red blood cell. I don't have it. And so that because of that's why I'm called blood group O, because I lack that antigen, that glycoprotein here. I don't have one. And because I don't have one, that is giving me blood group O. Okay, so these membrane protein can be used as well, can serve as well for cell-to-cell identity or cell recognition. Okay, good. Then another point, you can see from here that some of the protein here, okay, some of the protein, you can also see from here that, okay, so this is my first point here, okay. You can also see that some of the protein, look at this one. So some of the protein are also used for what? For transport. Transport. You see, we are talking about what? Channel proteins, okay? So these channel proteins, they are used for what? They are used for transport, all right? These channel proteins are used for what? They are used for transport, okay? Good. So in terms of transport, which one do we have? We can talk of what? They are involved in transport. So we can talk of channel proteins, okay, like the one you can see here. We have something like gate. We have gates and stuff like that, okay? But gates are also channels, so I won't worry you here. And then there's another we call the pumps. Okay? Channel protein transport carries out what? Channel protein they are used for diffusion. Okay, diffusion. Okay, diffusion. Oh, diffusion. Okay. So channel proteins are used for diffusion, so that when there is diffusion, when diffusion requires what? A channel, that type of diffusion is called facilitated diffusion. Okay, so when there is a diffusion that requires channel proteins, we call it facilitated diffusion. And then the pumps, okay, so let me put this one, put the bracket here. The pumps, this one is for what? Active transport. Pumps, they are, remember, facilitated diffusion, no ATP. And the pumps, they are what? For active transport. Active transport, so require what? Require ATP. Like that. Okay, they require ATP. Okay. Are you with me? So they require what? ATP. So the one that doesn't require ATP. So any transport diffusion in a cell that requires a channel is called what? Facilitated diffusion. And the one that doesn't, the one that diffusion that doesn't require a channel is called simple diffusion. And if it requires a channel, it is called facilitated diffusion. And then we have another protein structure here. Okay, with that one we call the pump. Those pumps, they require energy in the form of what? ATP. And so they transport molecules against the concentration gradient, which we shall talk about it later on. No, maybe not today, but we'll talk about them. Okay, so that one is for pumps are used for active transport because they require ATP. All right, good. And then another thing is that some of these membrane protein can also act as what? Enzymes. Enzymes. And remember, so some of the membrane protein can act as enzymes. And if it's enzymes, it means enzymes are what? They are biological catalysts. Catalysts. What are catalysts doing? They do what? Okay, biological catalysts. And what do catalysts do? They speed up reactions. Okay, so they speed up reactions. Okay, all right. And maybe one last thing that I will add is that okay, now most of the time in exams, if you separate channel protein for facilitated diffusion and pumps for active transport diffusion, I'll still mark you. Okay. I'll still mark you as separate points. All right, great. And then some of them act as what? Receptors. Receptors, okay? Receptors. Some of them act as receptors. So when we talk of receptor, what are we talking about? You know that sometimes even before the medicine we take, before it will get into the cells, there must be receptors there that's the chemical will come and bind and eventually get into the what? Into the get into the cell. Okay, and even pathogens. So receptors for not just so something receptors for what? For can act as receptors for viruses, drugs, okay, and other pathogens. Okay, pathogens, certain chemicals, okay, pathogens. Okay, do you know what? Some of us, there are a lot of people in West Africa. They can be infected by this. There's this plasmodium. We call it this plasmodium. We call it Plasmodium vivax. Plasmodium vivax can't infect about 90% of people in West Africa. Okay, the reason is that the receptor that the Plasmodium parasite will use, this Plasmodium parasite, P. vivax, the receptor that it will use to enter our red blood cells, about 90% of people in West Africa, they don't have that receptor. So because of that, we can be infected by P. vivax. There is this genetic disorder, we call it Crohn's syndrome. Crohn's syndrome. Am I getting it right? I think so. Crohn's syndrome. Like this. Okay, this Crohn's syndrome. There are some people HIV can be infected by HIV. At all. Even when you inject them with the blood. If you take every 100 people, and if you take 100 people, 3 of them can get HIV because of this genetic disorder. Okay, you call it Crohn's syndrome. What happens is that people with this syndrome, the receptor that the virus will use, okay, the receptor the virus will use, the HIV virus will use, okay, HIV virus will use, they don't have it. People with this one, they don't have that receptor. And because they don't have the receptor, they can't be infected by HIV. Okay, so receptors can be some of this membrane protein can act as what? Receptors for what? For receptors for viruses, for drugs, for some other pathogens, chemicals to get into the cell. Okay, some act as enzymes, okay, to speed up chemical reactions within the cell. And then under transport, we can talk of channels for facilitated diffusion and pump for active transport, and for cell-to-cell identity or recognition. This one is particularly for the for those glycoprotein or the surface proteins. Okay, so these are some of the functions of membrane proteins. So if I ask it, these are what I'm expecting you to write, not to tell me that they are for worn out. No, these are the functions of the membrane proteins. Okay, all right, good. So with the membrane protein in the bag, so let me just end with the functions of cell membrane, and then we can call it we can just give ourselves a break. Okay, so what are the functions of cell membrane? So let's see functions of cell membrane. The functions of cell membrane in general. Okay, great. So yes, one. Okay, good. Yes, anybody from the audience help me out. What are the functions of cell membrane? Yeah, it's when you talk of cell membrane, what is it? Yes, oh, today I've not called my ladies. Oh, okay, yes, where are my ladies? Where are they? Okay, anybody from the audience, please, what are the functions of cell membrane? Please, anybody should help me out. Somebody should help me out. What are the membranes? Yeah, it's with Pamucho.
It's the cell membrane. The cell membrane controls materials that can go in and out of the cell.
Okay, good. So cell membrane, I will use this way. Cell membrane, cell membrane is what? It is
Partially.
it is it is selectively
Yeah.
okay, permeable, okay, permeable. Oh, look at me. Permeable membrane, okay, and therefore regulate the movement of substances into and out of the cell. Okay, and we also know for sure that it encloses the cytoplasm. What else again?
So
What
please, a
else?
question.
Go ahead.
So when we talk of it's partially allows substances to move in and out of the cell. Substances like what are they supposed to be dissolved substances, or even if they are solid substances, they can still, move into and out of the cell, if I may know.
Let's take it this way that if it is selective, that's why I use the word selectively permeable. So the catchphrase is selective permeable. Let's take it this way that you have enough glucose in the word, enough glucose in the cell. It won't allow glucose to come in. If you have enough of something, like for instance, you remember SS days when we are organizing parties, we want girls to come. And so sometimes when you come and then a lot of boys are there in the party, especially the boys that we didn't invite them to come for the party, then the potato, we sometimes will become annoying because we want the girls to be there, okay, to the glory of God. You get it? So it's when you have enough of something, you don't allow more to come. Okay, so if I have enough oxygen, okay, I won't allow, if there's enough oxygen, I won't allow more oxygen to come. So today you can come in today, and then tomorrow when you are coming, you'll be stopped because you have enough of it already inside there. But if you don't have enough of it, we'll let you come. Do you get a difference now? But
Yes,
for
please.
fully permeable membrane like cell wall, that one everybody goes to. Everybody goes. Right? Everything can enter. Right? But this one, it only allows certain substances to enter. Today, to allow you to enter, the following time, it won't allow you. Okay? So the following time is like, no, we have enough of this. No, we won't allow you to come. Okay, great. So that's what makes it selectively permeable membrane. All right, because you need to control the internal environment. Things must be stable. So if I have enough sugar in those cells, there's no need to absorb more sugar. Because when you absorb more sugar, it's going to be disaster. So the body will be controlling all those things. Okay, if you have enough glucose in the cell, then the one that will be absorbed will be converted into glycogen. Okay, all right, so
Okay.
that
Okay.
it wouldn't create problems. Okay, so that's what we refer to as what selectively permeable membrane. It's just selective. Today it can allow you to come, tomorrow it won't allow you to come. Okay, all right. Now, but if you take something like animal cell, in animal cell, you realize that it is the what? It is the outermost layer, okay, and therefore protects what? It protects, support, and gives what? Gives variable shape to the word, to the cell. Okay, all right. So these are two general functions of cell membrane. Okay, so being selectively permeable and in animal cell, okay, protect, support, and gives what? Gives variable shape to the cell. Okay, all right, good. So I will end here so that if you have any, you can give yourself a break. Okay, all right. To me, I think we've done a long haul. Let's kill it. Maybe let's kill it to maybe 45 so that when we break, then that is it. I'll put questions there, then we solve it. When we break, then we don't come back. Okay, so let's kill it. Let's kill some 20 minutes more, and then because we've gone a long haul, and you've been with me all this while. You guys, oh Charlie, I know my ladies are tired. I can see the tiredness. Oh, Angie, I can see the tiredness. Oh, sorry. Sorry, please. Mercy, I can see it. Charity, please, I do see. I'm sorry. Please forgive me. Okay. Let's just kill. Let's kill. Let's kill. Okay. Okay. So, that is about cell membrane. Okay. All right. So, remember, I made mention of cell wall as well. You know, cell wall in plants is cellulose, mainly cellulose. In bacteria, remember, is peptidoglycan. We mentioned that. And in fungi, that's the mushroom and co is chitin. Don't forget that. Okay. All right. And those are non-living components of the cell. Okay. So, okay, good. So some of them I've already talked about them already. I wouldn't waste your time on them. Okay, that's much. So what we're gonna do is that we'll move on straight to organelles. Okay, and then we'll run through the organelles quickly, and then we can call it a day. Oh, you guys are tired. Do you need a break now? Do you need a little bit of break now, please?
Yes, so that we
No,
can
sir,
go
we
and
need
come
a
back.
Yes,
Oh,
Yeah, I
please.
know.
We
I
need
know.
a break. We need a
Yeah, I
break,
know.
there should be a
please.
break. There should be a break so that we
Yeah,
can go and come
Let's
back,
I
finish.
could
Let's
see.
finish it.
I could
continue,
see. I
and
could
Yes,
see
then.
the
so
ladies are
Uh,
we
tired.
need a
No
break.
problem. No problem. Oh, my ladies, please, I
Your
will do
girls,
that.
your ladies are tired. Your ladies
I
are
know. Please,
tired.
your wish is my
We're almost
command.
done. We're almost
Your
done.
wish is my command. Your wish is my command, please. Once the lady is
We're
tired.
almost done.
Right.
We're almost done. Let's kill it. Let's kill it. We're
Oh
almost done.
no. The ladies are tired.
Oh,
Please, the ladies are
no,
tired.
no.
My
Yeah, we're
Don't
ladies
be
are
almost
as
tired.
done. We're almost done.
tired.
Let's finish. Let's finish.
My ladies are tired. Okay. So let me just give
Let's kill it.
you
Let's kill it. Let's kill it.
oh, please, my ladies are tired. The ladies are tired. So that I will also turn and make some small cup of tea.
All right. All
You
right.
think
Yeah,
the
you have
break
to
is
go
for
for it.
you alone? Son of man need to not let you say alone some tea and bread to also to the glory of God. You know what I'm saying?
how many minutes? How many minutes break, please?
Okay, please, this one we don't have to waste too much time. So let's give ourselves
Oh,
like
11 o'clock is up. 11, 30 minutes.
No, 30 is too much.
30 minutes to be too much. Do you have another lecture? Please, do you have another lecture?
Yeah,
Do you
No.
have
yes,
another
Yes,
No.
lecture,
please.
please?
Yes,
Yes, sir.
Yes, we have another lecture.
chemistry
Yes,
major.
chemistry
Uh-huh.
people.
Please, it starts at what
Also
time?
for
The chemistry
chemistry,
starts
they
at what
can leave
time?
so that you will continue.
Hey,
I
no,
think
no, no,
11,
no,
11
no,
30.
Oh,
no.
please. Oh, please.
11:30,
Oh,
like.
please. It starts at 11:30. So then please, if the chemistry people start at 11:30, then please let's make it the thing like 15 minutes. Okay. All right. 15 minutes, please. This one that
No,
I'm
we
begging
start at
you,
11.
please.
So 10:45.
It's 11:15
Yeah,
rather.
so like so let's say we are coming
10,
back at
10:45.
10 for
45.
no not 10:40. Okay, 10:45, basically.
Yes.
10:45. Okay, 10:45.
Right. Thank
Okay, great.
you.
Great.
Your
Thank you.
ladies are very grateful.
Oh, well, it's so sweet. Thank you. I will do everything for you. You know, I will. I will. I will. You are the reason I live. Okay, I live for you guys.
Yes, yes, yes. I will love you for that.
Oh, thank you. My day is completed. With those sweet. The New Year will come on what? The New Year should be on Thursday, but mine has started today. Without sweet words coming from my ladies. Oh, okay. Thank you so much. All right, so let's give ourselves a little bit of break, okay, and then let me get some tea, and then I'll come and type some questions for you.
Thank you, sir.
I can do it while we should. Let's enter the field. This is it. Also to understand I have to do what so I don't know around the rest of the maybe one day we will get to pay him.
My people, are you in? Have you started? The men, have you started? Or your wives are teaching you? Yes, shame on to you. Those who feel that your wives are teaching you, I've seen you all.
Doc.
After that, come and tell us.
You have provided our answer to the men we are on.
I want you to know that God has seen you. Peter, God has seen you. Peter, you are there. Your wife will teach you. You solve the two together and come and tap 25 for here. You will see what will happen. Today, it's like it will be war. It will be war. Peter, woe unto you. Woe unto you. Thou say the Lord. Peter, I say woe unto you. Thou say the Lord. Your wife is teaching you. I saw you feely. Teaching you. Woe unto you. If you woe unto you, and you want 25 on this corner here. Charlie, Supreme Court with pink sheet all will come inside. You
and I were not there.
Me, Peter, I say I saw you. Peter, I saw you. His camera was on, and I caught you feely.
Please, I just 2 questions.
Yeah, there are 25 questions, please.
Please, kindly turn the other side.
Okay, so please, 25. Okay, let's see. Let me go to the, okay.
So wait for a minute
Okay.
before you turn it on.
Okay, maybe you can screenshot it. You can take a screenshot of it on your phone. Okay. Please hurry up. Our chemistry folks need to go to class, so we don't have to take all their time. Peter, don't write any answer here, because I caught you feely being taught by your wife. Mary, your hand is up. Rachel, your hand is up. Your hand is up. Okay, I will switch on to the. Okay, a few minutes, and then we can call it a day. But on more serious note, we need to look for extra 1 day somewhere. Let's take it. Don't get to your Tuesday. I'll meet you. Okay. Can we meet Wednesday evening? Oh, Wednesday is 31st, so that's impossible. Okay, people will be busy preparing for. Can we meet 31st in the morning? 31st in the morning. It's no holiday. Hello, can we meet 31st in the morning so that we can get some 2 hours to be? Let's see if you can get 7 to 10 on 31st morning before the chair. Yes, so that
No problem.
you can see that I am moving on slow pace so that you get all the concepts right. So
if possible.
yes, yeah, sure. So I think with that one, I will speak to Becky about it so that she can set it up for us on 31st night. On 31st, okay, early in the morning, maybe 7 to 10, and then so that we'll clear things up, okay, so that we'll be able to finish. Okay, once we get one more meeting in between, we'll be able to finish that. Okay, yeah, okay, good. All right, so 1 minute more, we should be done, and then we'll answer the questions together. 1 minute more, we should be done, and then we'll answer the questions together. Okay. Yes. Yes, okay. All right. I think we can solve it now. We can start solving it now. Do you need more time? Just 1 more minute, and then we start solving it together. Okay. Okay. All right. So let's start solving it together. Okay. So number 1, All cells have ribosome. True or false? Yes, number one. All cells have ribosome.
True.
It's true
True.
statement. Good. Number two. During organ transplant, during organ transplant, the compatibility of the glycocalyx of the cells must be considered. True or false?
True.
That's true.
True.
The shock of glycocalyx. Please, if you have noise in the background, please mute it for us so that we can be snappy. We have words. So here, glycocalyx is actually referring to those glycoproteins. Okay, all right, good. So this is true statement, and then that's true. Then the next one, unlike food vacuum, contractile vacuum is not permanent.
False.
It's false.
True.
Okay.
Oh, false. Okay.
I heard one man say true. So
Yeah, one man down.
one man down, one man down, one man down, one man down, one man down. I don't want this one. I don't want any. I heard it loud and clear. Please, I heard it. One man down, one man is down. We need back up. Facilitated diffusion. So both are all temporal vacuums. Okay, so facilitated diffusion requires ATP. True or false?
False.
False. That's good. Well done. Well done. Well done. That's false. Well done. Endosymbiotic theory was proposed by Theodore Schwann and Matthias.
Yeah, you listened to.
Oh, sorry. It's
So for the
false.
question
It's-
4,
sorry,
you
sorry.
answered 2.
Endosymbiotic theory was proposed by Theodore Schwann and Matthias Schleiden. True or false?
It's false.
False.
It's false.
False.
Yes.
Correct sequence. The rate at which the men will allow the lady to say it and they say it after the ladies. I don't like it. I don't like it. Please.
Oh.
Please. It's because of you that God nearly spoiled the world again with flood. I don't know being the intervention, intercessory prayer of Noah would all be drowned by now. And men, what you are doing, I don't like it. Okay. You allow the lady to say it and you say after them. I don't like it. Please. The correct sequence of the cellular respiration is Krebs cycle followed by glycolysis, followed by electron transport chain. True or false?
False.
False.
False. Good. It should be that. It should glycolysis first and then good. Next one. Glycolysis cannot occur in the presence of oxygen. This question is tricky.
True.
True or false?
True.
Oh, one man down again.
It's false.
It's false.
So it's false. Yes.
It is oxygen independent. It is oxygen independent. So to occur, whether oxygen is there or not. So another man down. Second man. Second man. Dead.
So what if the ladies go?
No, I didn't hear the lady. I'm listening. I listen attentively to ladies very well. Ladies very well. Even those people, those men who are saying this is because their wives are standing behind them. Because I've seen Peter. Peter is the clear example. So,
Our wives are giving us wrong answers.
yes, good. Primary cell wall.
Our wives are giving us wrong answers.
Primary cell wall. Let me read this one. Primary cell wall is thicker in plant. Is thicker than the secondary cell wall. True or false?
False.
False. Good.
False.
The ladies in this class, they are something else. They are something else. Next one. When developing antibiotics, the best organelle to target in the bacteria is the mitochondria. True or false?
False.
False. Mitochondria the bacteria doesn't have mitochondria. So why we have so what's the best one? If we're to be the one developing, what's the best one to target?
Plasmid.
Nucleus.
They don't have nucleus, so you can't target nucleus. You can't target plasmid. Good. What else again? Peptidoglycan. Good. That's excellent. Plastic peptidoglycan. What else again? And then you can target ribosome.
Okay.
The ribosome in the prokaryotic cell is different from the ribosome the eukaryotic cells we have. Okay, so you can target. So well done. Plasmid, peptidoglycan, and then the ribosome. Well done. Mitochondria, nucleus, Golgi body, and chloroplast are all products of endosymbiosis. True or
false?
Why is it false? You see this one, the men are not talking.
Yes.
The men are not, they are scared. You will keep quiet. I know. When it becomes tough, only the ladies speak. Oh God. We should make it ladies class. We should name the class ladies class. I'll tell you. The class is for the ladies. You see, so this one, only the mitochondria and the chloroplast are products of what? Hey, who is talking? You don't,
Angela.
The lady.
Ladies are yes. It's just translating the science to the men.
No, I mean like
One lady down.
Next one. Plant cell with an even thickness of the cell wall is most likely to be sclerenchyma. True or false?
It's false.
Okay, that's the vim. Good. Who said true? Another man said true, Randy. Hey,
Lady.
He said
oh, yes, one man said false, another man said so. One man down again.
No. I said false.
Oh, you said false. And I heard another one say true. Eh?
The man
was a lady.
No, it's not true. No, the ladies didn't talk. They didn't talk. So yeah, sclerenchyma. Yes. Cholesterol is an amphipathic molecule. True or false?
It's false.
It's true. It's true. It has both polar. It goes through the entire length of the cell membrane. So the head regions, they are polar. So it will be
one
there.
man down.
One man down again. One man down again. Oh. One man. The men, they are dying, Papa. Oh God. Prokaryotic
I heard a
cells.
lady also saying false.
Oh, a lady.
Oh, thank you.
Yes.
Oh,
It's a lie. It's a lie. Big lie.
Oh,
you live long.
I didn't hear. Oh, I didn't.
No, you didn't hear. It's a lie. You didn't hear. It's a lie.
Thank you. Prokaryotic cells lack organelles. True or false?
False.
Hey, what is happening to the lady?
One lady down.
It is a slip of tongue. So it's a slip of, I can't write. It's a slip of tongue. Okay. Yes. Like the way I was writing through and I wrote it's like that. It's a slip of tongue. The cell wall of bacteria is made up of chitin. True or false?
False.
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 Look at the way the ladies are saying it with them. Then the next one, the opening and closure of stomata are controlled by the gut cells. True or false?
True.
Lysosome and ribosomes are non-membranous organelles. True or false?
It's false.
It's false.
It's false.
The lysosome is membranous organelle. No bacteria can perform aerobic respiration. True or false?
It's false.
It's false. They can use the cell membrane. Okay? Next one. A secondary cell wall consists of cellulose, lignin, and pectin. True or false?
False.
It's false.
Yeah, they don't have what? They don't have pectin, okay? So that nucleus, Golgi body, mitochondria, chloroplast, all form part of the endomembrane system. True or false?
Oh, it's true.
False.
It's false. The membrane, yeah.
False.
It's false. No. Now, do you know what? Mitochondria and chloroplasts, they are not part of endomembrane system. Even though they are membranous organelles, all right, but they are not part of the endomembrane system. Okay, next one. In animal cells, excess glucose is stored in the form of glycogen. True or false?
True.
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 I know. The ladies will say it. I know. Fast. Chemistry people. Mitochondria is larger than ribosome. True or false?
True.
Fluid mosaic model was proposed by Singer and Margulis in 1972. True or false?
False.
False.
It's false. It's Singer and Nicholson. And then peripheral proteins can be transmembrane or not.
False.
It's false.
It's false. Okay, it's actually integral protein.
Integral protein. Rather integral.
The head of phospholipid is hydrophobic due to the presence of phosphate. True or false?
True.
It's false.
Another man down again. Look at the way he said it through with him. Oh God. One down and
false.
Look at the way the man who said the truth, the way the zip is used to say it. Okay, you see, all right. The zip, he fell down on the battlefield.
The radar is hydrophobic.
It's even nice. The zip is even high. Okay, so please, the head of phospholipid is hydrophilic. Okay, the head is hydrophilic. Okay. Then all eukaryotic cells have mitochondria. Wow, this question is tough one. True or false? Hey, the medicine. Look at it. Yes, come. Yes, the man. I want to hear you. I want to hear the bass. Hurry up!
It's true. True statement. It's true. True statement.
Red blood cell. Red blood cell doesn't have mitochondria. Mature red blood cell doesn't have mitochondria. Okay.
Yay. One man down.
Oh, shit. So another man down.
One man down.
All right. That's another one. You know that they know themselves. All right. Okay. So who actually 5 over 85? Who are 5 over 85?
No, sir, doctor.
Anybody with it? Yes, please.
Yes. With the question 25, I would wish you show more light on it.
Yes, not all eukaryotic cells have mitochondria, like red blood cell.
Yes.
Mature red blood cell doesn't contain those membranous organelles because it doesn't even contain nucleus. In place of the nucleus, that's where the hemoglobin is located. So not all. I said all eukaryotic cells. Okay, but that's not true. So that's what when we meet again, we will do about specialized cells. So specialized cells, some of the organelles are missing, some of them are mini and stuff like that. So this statement, because I brought all eukaryotic cells, but that's not true.
I was looking at the diagram of the plant cell, the animal cell.
Yes, that one is generic structure. Structure. Okay, but we shall do the specifics. You see that, for instance, have you seen that? Let's say cassava, like plant roots doesn't have chloroplast.
Okay.
The cells of plant roots, because they are not going to absorb light for photosynthesis. So even though their plants are all right, but they lack chloroplast because they are in the soil. And in the soil, the sunlight doesn't reach them.
Okay. All right.
Thank you very much. So please, is it a deal that we are meeting on Wednesday in the morning?
Yeah.
I will note So we'll be able to finish. Okay. Thank you so much for having me.
You too.
And then I wish
Please,
you
the
all the
slides.
Thank you
have
so
a
The
much.
slides. When is it coming?
Nice.
We are grateful for you.
as
My men will not be down.
The ladies
Are
are
the
very
slides on the platform?
nice.
Yeah, so I'm going to send it to your colleagues. I'm going to send the slides to your colleagues right now.
My men will not be down next time.
Okay.
Doc.
Thank
Hello,
you.
Doc.
Yes, please.
Please, the other recordings, we have not gotten the first recording yet for cytology and genetics. I don't
I
know if
think
you
the
can.
first one that we had, we couldn't record it because the link didn't really work well. Okay, so we couldn't really record it. So, but please just read through the slides, and then if you don't understand anything, feel free to WhatsApp me. I'm always there for you. Okay, you know you are my lady too. You are the reason I leave. Okay, so please. Okay, once you don't understand,
all right. Thank you.
then I will WhatsApp.
The man, let us ride.
For the man, for the man, when you WhatsApp, I get to respond.
It will be there for a long time.
The first recording I have.
It will stay there for a long time.
Yes, to be foretold is to be forewarned. For the man, when you WhatsApp, sometimes it can take 3 years before the reply will come. Yes, we have told you in advance. Yes, please.
The first.
It doesn't know. Is it not
yes,
simpler
please.
than?
Yeah,
yes, please.
The first lectures. I have audio recording of it. It is on our Telegram page. You should go there. They will get.
Oh, okay. Oh, then please make it available to my lady. Say, you see, the first one.
Then share the link on the page so that we can join.
For my lady. Okay.
Share with the ladies.
First, give them that.
Yes, please.
Say, not only the ladies, so we are all involved.
Yes, I don't care.
All join the Telegram.
Share the link.
So please share the link with them for me.
If what is saying that it's a must. So share the link.
Okay, I'll do that pretty soon.
Yes, don't
all
give
right.
it to
So
the ladies,
I will share the slides with you very soon. Okay, so that
we are waiting for the slide.
Yes, I will do that. I will do that. So,
okay.
Thank you. Have a nice day, and God richly bless you.
Bless you too, sir.
Thank you.
Yeah. Kafui.
Yes. I'm here. Somebody called.
Please, we don't have a note on cell and tissues organization, so can we get it?
You are who?
For me, I don't have it. I'm PD if I can get it.
You don't have it because I'm